Richa, I believe we can begin now. It's 10:30.
Sure. So I hope, Elora ma'am, you are there online with us now.
Hello.
Thank you, ma'am. Good morning.
Good morning, ma'am.
Good morning. Good morning to
And
all of
welcome
you.
to Meeting Online program, ma'am.
Thank you. Thank you very much. I am also proud that you have asked me to join such a program you are conducted.
It's our pleasure, ma'am.
Yes.
And we are very thankful to Juhi that she brought us with us, and we get an opportunity to listen to you, and we are looking forward for your deliberation.
Okay. Okay. Thank you. But please help me out how I will open my presentation here.
Yes, ma'am.
Because I am not so much accomplished with all those things. I'm sorry.
No, ma'am, it's fine. Ma'am, आप अपना presentation हमसे share भी कर सकते हैं, so somebody from here can also share your presentation. Otherwise, our IT team would guide us how to share the presentation at your end.
But Dr. Ambina, it's better if ma'am send her presentation and we will share from here.
Okay, fine.
Ma'am, please send your presentation.
Okay, just a minute.
And to whom you are sending your presentation, ma'am, so that accordingly we will share.
Anamika Srivastav, who has sent me.
Okay, ma'am. Anamika,
Good morning, ma'am. Yes, ma'am, we'll do that.
Okay, I have already sent it.
Just checking, ma'am.
Just check it out. But it will be better if I can manage it. So probably it will be not possible.
No, ma'am, it is absolutely possible because you'll take time, while speaking, which slide you have to emphasize more, which you have to.
Yes.
Ma'am, आपकी
That if it is not possible from my side, then you will reload it.
Okay.
I think that would
Okay,
be
ma'am.
better. So
Ma'am,
now tell me
uh,
how to do that.
Yes, ma'am. मैम आपकी स्क्रीन पे शेयर स्क्रीन करके ग्रीन कलर का बटन होगा जूम की स्क्रीन पे.
मीटिंग इंफॉर्मेशन ग्रीन कलर का है.
Arvind sir, can you help us with this?
Share screen. Okay, I have
Ji,
got it. So
Okay.
I am sharing it. Share. Okay. It's
Yes,
all.
ma'am.
Now I can.
Yes, perfect, ma'am. Cadmium toxicity in mice model biochemical proteomics and metagenomics analysis. We can see that, ma'am, and then you can put it on slideshow.
Yes, I have already put it on slide. So I think it will be visible to you people all.
Yes, ma'am. But you have to reshare it because before that we're going to play a video for you, for a small video for felicitation. So yeah, please reshare it again.
So what I will do now?
कुछ नहीं मैम, आप प्लीज अपना
Ma'am,
presentation है.
can you please stop the sharing for a while so that we can begin with the proceedings first, welcoming you formally, and then you can please share it again.
Okay.
Thank you so much, ma'am.
Thank you.
May we begin?
Yes,
ma'am?
Richa, please.
Okay, a very warm welcome to all our distinguished speakers and participants. Good morning, everyone. I, Dr. Richa Nagar, on behalf of Amity Institute of Environmental Sciences, Amity University, greet all our esteemed speakers and participants to the fourth day and plenary session 6 of our 5 days training program on management strategies for environmental health and safety, a step towards achieving SDGs. From 16th to 20th January 2023, in association with Indian Institute of Toxicological Research, CSIR. Health and well-being for all at all ages are at the center of sustainable development as determinants and enablers of the SDGs and as outcomes. In order to effectively control factors that may affect health, the requirements that must be met in order to create a healthy environment must be determined. Environmental health focuses on the natural and built environments for the benefit of human health. Therefore, today's sixth plenary session focuses on environmental pollutants and monitoring of health effects. With the experienced panel of speakers, we are sure our participants will gain a lot of insight into the topic. Our first speaker today is Dr. Elora Ghosh Mondal, Associate Professor, School of Environmental Sciences, JNU, Delhi. Her areas of interest, specialization are environmental toxicology and human health, cellular and cancer biology, clinical biochemistry, application of biotechnology, biochemistry, and molecular biology in the field of stress biochemistry, cancer biology, and reproductive biology. With a vast experience of over 2 decades, she has many awards and honors to her credit, along with several high-impact research publications. We are honored to have you with us today, ma'am. As a tradition, we would like to felicitate you with a virtual Tulsi sapling plant. And for that, I request Mr. Naresh to please share our traditional Tulsi sapling. We welcome Dr. Elora Ghosh Mondal, Associate Professor, School of Environmental Studies. So I would like to hand over this virtual Tulsi sapling to you, which is our tradition offline and online. Thank you, ma'am, for joining us. We are very eager to hear from you, ma'am. Elora, ma'am. I think she's facing some technical issue there because her mic
Dr.
and video
Juhi?
is on, but
Juhi, please contact Elora, ma'am.
Yes, ma'am, I'm giving her a call.
so I think she's joining back again.
I think she's joining back.
Ma'am, could you please unmute yourself?
Suddenly connection has been lost.
That happens, ma'am. We are facing the online world since the pandemic, so we are used to it by now that these glitches come on and off. So welcome, ma'am. Welcome to you. And
Thank you.
we are looking forward to your gracious lecture here.
Okay. Now I will. Is it okay?
Yes, ma'am, absolutely fine.
It is not changing.
We are on the title slide right now.
Okay. Okay. Good morning to everybody. I am honored to be along with all of you, all participants, as well as my dear colleagues from esteemed Amity Institute. I also think whatever I will tell may be appreciated or related to your link of presentation on that aspect and introduction. I will start with cadmium toxicity, and we utilized in our laboratory based work. I am giving here on the basis of some mice model activities only. Also some cell culture works has been already done, but I won't give those things because it is a management study, management related things. So I think it will be preferable and appreciated by all the participants. So my topic is cadmium toxicity in mice model and its approach in biochemical proteomics and some place of metagenomic analysis, but on a base of primary articulations, I must give some information related to cadmium, how it comes to our body. When we are thinking about any of the metallic component, it is not easy to enter within our system. I want to mean system as interior, not exterior. I may take plenty amount of mercury, but mercury cannot be absorbed as it is. I may take iron, but iron cannot be absorbed directly to our body. So for that, some biochemical processes are needed, or some specific organic or inorganic component, and attached with that those so-called heavy metals are entered inside our body. So it is not that if it is present, it must be inside. So if it is so, then from the long ago it will be. But nowadays we are talking about pollution, more pollutant, and all those stuffs, and that means it is such a condition that we engulf it as well as absorb it within our system. So from weathering of rock is coming down from the day of earth process. So from there it is coming, but now it is problematic because the wash off of those weathered rocks are coming to our drinking water process or something like that. Forest fire, it came us by air pollution or pollutant. Not only for a forest fire in Delhi, we are facing lots of other paddy fields for fires and all those ashes as a PM 2.5, 10, and all those things. Volcano eruption, of course, surrounding India is very little, very less. But in whole world, it is coming also for that thing, from that places. Metal smelting and refineries, all the metallic plants we are getting. Mining, we are getting plenty of cadmium containing mining processes. Surrounding which we may have these type of contaminant, and thermal power plant is very less nowadays. But even after that, though protection and processes from the government of India has been evoked to them very strictly, but the very nuisance which are from our side only. Actually, human being, as a human being and as a third world country, first world country, second world country, whatever countries are, we are throw out our old electronic waste as a debris anywhere else, and they contain huge amount of all those places are containing cadmium. But just like mining wash of water, electronic waste waters, these are coming very quickly, very efficiently, so-called efficiently, as in our drinking water process, or any of the, even some places groundwater are also contaminated with cadmium. But the flow of water is always contaminated. If you go for identification or estimation of cadmium, you must get a trace amount of cadmium. How much cadmium is present, that is a question, and below that we are very happy to accept it. Now I want to give some ideas that if it is below the level of contaminant, the limit of lethal dose, whatever we may take from the analysis and estimation from the chemical things, but this is the nuisance of our knowledge, that we are safe, but we are not safe. So that is the space I want to access or express my concern to all of you. We know that 1.2 billion people in the world is not accessing that type of safe waters, drinking water, and 2.6 billions are not have access to clean water. So you understand how much, and many of them are surrounding in India also, outside, abroad also. I am not denying it. But the estimate lethal oral dose, whichever I am telling, 350 mg of cadmium. And 350 mg per liter of cadmium is a very, very huge concentration. So we are 100% safe, 100% safe from cadmium toxicity. So we are not concerned about cadmium toxicity. Okay, but then why I am speaking today about cadmium toxicity? And it has been found that 3 mg of cadmium is not at all affecting in the system, human body system. But I can show you how it can affect with a cumulative effects and with a change in the biological system within ourselves. And we are gradually encroaching our amount of level of amount of heavy metals limits within our body system, and that's the most crucial factor in our concern. We know Itata is a disease which has been very much famous, and for that reason only today we are talking about cadmium. Maybe some other diseases have not been identified with some of the specific heavy metals. So we are not concerned about selenium or other component, but after a few days we may get some information, and then we also concerned about. But when Itata disease has been identified and going for those pain and recovery and all those stuff, then people even not imagine that cadmium may be a contaminant, and it may come and substitute our calcium in the bone, and so the bone has been very much fragile and going to contaminate, going to give us so much pain with that contamination. So this acute cadmium toxicity we are very common with all other heavy metals, जैसे कि nausea, vomiting, diarrhea, these type of abdominal pain. These type of problems are very, very common, not only with cadmium toxicity, but also with various toxicity. Now I will come to, and I will put one point before that. The first identification of cadmium toxicity has been happened to be identified within the lungs component of lungs has get a huge amount of cadmium, and due to this, along with some other component or other heavy metals also, it has been identified to induces carcinogenic. Actually, it has not been identified as carcinogenic component, but it has been identified as a component which can destroy the proper absorbability of the lungs system. So there is a char formation, and there is a cirrhosis, this type of aberration, and blocking, choking, these type of things. And it has come from the smoke, maybe cigarette or maybe other things. So people are cautious about their mask, and maybe they leave their smoking. But around 2005, 2006, it is predominantly identified that cadmium as a pehle ऐसे नहीं, people cannot identify it in such a way, but now it is so much so that in the water system, cadmium as cadmium chloride, cadmium silicate, and some other components, it is very much present. So that's a huge problem nowadays. That and it is not easy to identify cadmium in the water system, even in the drinking water purifiers, as some of the water purifiers are try to give some addition of plus to bivalent ions. So for them, the cadmium may be a leak within the drinking system, even after the purification. So cadmium is very crucial because it has that capacity to into some places. If it is very high in amount, substitute the calcium, that is the very crucial bivalent cation in our body system. All the system in the total mitochondrial system has carry on with plus ions, calcium ions, but cadmium may substitute if the concentration is very high. So it is also very important to inform you people that this cadmium is a component which has the capacity to stay within my system. So I am very much curious about I should not take cadmium or calcium or iron or something. Is not the toxicity. What is toxicity? Toxicity is there starts there where the concentration is above the level of the component must be present within that territory. So it may be within a cell, the concentration is something, within the serum is something, or then the organs are different. If it is higher in liver, maybe it is less, must be it is less in cardiac tissue or kidney or lungs. So that is also a very important phenomenon, that any of the component which has the capacity to go within the system and circulate, distributed throughout the system, the distribution peripheral and internal must be different to accommodate. It is not that all the organs can accommodate huge amount of different amount. They always differentially react. So that is another concern I will tell in my after few slides. That is another thing is it generally deposited in play. All the metallic components are generally deposited in the kidney. So kidney stone is one of the source of cadmium. And other thing is it has been shown by various people in various journals, but the actual process of incorporation, actual process of interaction is not known very clearly. Still now it is not known very clearly. Some of the papers have come afterwards, but it is not very clear identification. But it has been found that cadmium has that some role in cardiovascular diseases, in cancer diseases, in neurobiological diseases, but where and how people don't know. But it is confirmed any of the bivalent cations like cadmium induces stress, oxidative stress generally within in vivo and in vitro system. But here I can show you a good presentation where you can see the cadmium has been absorption. Any component may come within our body, but may not, the total percentage has been absorbed by the system. Whichever present in the outside the environment is not absorbed by our body system. Some of the body system is much more competent to reject it. Some of the body systems are physiological systems are so part of that they won't give them even entrance within the system. Some have absorbed and degraded and extracted in a proper way. But every time, if there is a very acute amount of component, X component, any toxic component, ऐसे नहीं है कि कोई भी अननोन कंपोनेंट को बॉडी नहीं ले पाएंगे, देन तो कब तक हम लोग मर चुके थे. It is not like that. The level when the body cannot work, then it must be deposit. अगर आपके घर में बहुत सारे सामान इकट्ठा आ गया, what you will do? You will keep it in front of your door and आहिस्ता आहिस्ता थोड़ा थोड़ा करके ले जाके proper जगह में रखेंगे. तो it is something like that. Oh, they will preserve it generally around liver. Firstly, they try to throw it out by the kidney. So some of them are deposited in kidney. Some more of the part has been deposited to the liver, but or lungs. But after that, they cannot do. Then what happens? They so much things cannot be controlled. So different components are there: protein, carbohydrate, fat. कोई भी एक कंपोनेंट के ऊपर वो भी एक केमिकल है, वो रिएक्ट कर ही सकते हैं. एंड कैसे रिएक्ट करेंगे, दैट इज द क्राइटेरिया. व्हाट इज द टॉक्सिक इफेक्ट्स यू कैन विजुअलाइज एंड हाउ यू कैन टारगेट दोस पॉइंट्स. सो फर्स्टली लंग से एब्जॉर्ब होते हैं. इफ इट इज थ्रू लंग्स तो कैडमियम सिस्टीन से ही होता है. सिस्टीन बाय बॉन्ड को ब्रेक कर देते हैं, एंड दैट मीन्स व्हाट हैपेंस, प्रोटीन हैज बीन डिग्रेडेड. How, from where it can get the cysteine? It will get the cysteines from nothing other than from. I'm sorry, may I ask what is the time I have? 10:15.
Yes, ma'am.
Ma'am, it is up till 11:15.
11.
You have
1:5.
yes, 11:15. Till 11:15 you can continue, and then we have 10 minutes question and answer. So you have 20 minutes to go.
Okay. So from there, this actually I am elaborating some introductory part, probably more. Anyway, this is also a good information, I think so.
Yes,
so
ma'am.
absorption of cadmium, from where it can get the cadmium? It must get the cadmium from cysteine, and it is bind with the cysteine. But from where cysteine will come? Cysteine will come from only from the proteins, and the proteins which have a much more amount of cysteines, they breaks, and if it breaks, the protein is degraded. So ultimately what happens? Some of the proteins has been degraded due to cadmium toxicity. So cadmium has that capacity to do so. And when it has been charged positively, then only it can react to it. So अगर only cadmium दिया जाएगा तो कुछ नहीं होगा. If it is in a hydrated form, मतलब cadmium chloride, generally the source is cadmium chloride, cadmium methane chloride, some other organic component के साथ जोड़ के भी आ सकता है. So acid condition में oxide, generally cadmium oxide is a very fragile component, so it generally not sustained. It always breaks and go for some of the proteins or carbohydrates or something like that. And cadmium methylothionein बोल के एक protein है within the biological system, मतलब any animal system, that methylothionein has only the work to attach with the metals. Mostly bivalent cations. And agar wo bivalent cations ka attach karenge, it is a very good component. It has the capacity to carry it on to the kidney and release it to the kidney so that it will excrete it, glomerulus, so that it will excrete it from the system. But agar tum bahut sare cadmium chloride le liya, if you have engulfed so much more amount at a time, then how much cadmium chloride metallothionein will be there? Nothing like that. So it will degrade, or some other components will degrade. So it is interact with them in liver. Cadmium, not cadmium, any bivalent or more divalent cations and trivalent cations ka degradation process hai. There is a process of metabolism, and this metabolism process helps with metallothionein, glutathione, any cysteine divalent proteins, and any conjugated things they attach, and generally they push it to the biliary system, and after that it goes to the same to the kidney, and from kidney it will excreted out. But agar wo jada ho jayega, same as gastrointestine, same as kidney, excretion is the process, but agar wo jada ho jayega, to no control, so it will goes to the serum. And when it is within the serum, we can actually, if we taste the urine, we can get the identification how much contaminant are there. If we go with the serum identification, we have to go for some of the enzymes activity, and from there we can identify what are the components. So by which recent, I won't say recent, but 2016, it has been found that cardiovascular disease के लिए बोला गया है कि cadmium is a toxic component for and can induce cadmium cardiovascular diseases. But how it is induces, it is not arguments are there, but it is not established fact. Pulmonary disease is already established fact. Everyone knows kidney loss function is also been found with higher level of bivalent cation toxicity. Gout is one disease has been identified with cadmium toxic level is high, cadmium component is high within the body system. Bone loss is the identification, that is the Itata disease has been found. But now it has come to our vision that any bivalent cation such as cadmium has a variety of pathways from where to where it can go. It may go apoptosis. Apoptosis is a pathway of process death. Means if the cell cannot control, they may burst out and spoil. That is one process. जिसको necrosis भी बोला जाता है. The other process is apoptosis, where it is giving the sense to the surrounding cells also that something is abnormal. So these are the signals. Be careful. Either you can overcome it or you have to leave it. So this is the process of apoptosis. And another is basically adaptive response, how we can recover it, how we body can, ऐसे नहीं है कि कोई भी toxicity हुआ तो तुरंत मर गया, वो तो नहीं होता है. So there is an adaptive system. The body can adapt, absorb and restrict it in such a place or point where it can survive. There will be no problem. Other places is some of the folding, unfolding. That is the place where the necrosis has happened, uncontrolled. System cannot control it, cell cannot control it, organ cannot, and that has come to be a cirrhosis. At that sense, that is a abnormal situation, and spots of passes and all those bacteria may grow, and some other problems may arise from that part. And in some places with a very high, maybe 300 mg and more than 300 mg concentration, it has been detected as a cancerous cancer injury, as in the kidney it has been found, detected also. But all those things has been found to be regenerated by the oxidative stress damage of different, जैसे मैंने बोला cysteine divan को break कर देगा, कैसे कर देगा? This oxidative stress can induce this type of phenomenon. But सब कुछ खराब नहीं होता है, उसमें कुछ अच्छा भी होना चाहिए. So I will add on something that there is a treatment of cadmium toxicity. Generally क्या करते हैं, अगर बहुत सारे कैडमियम क्लोराइड तुम पी लिया तो तुमको कैसे बचाएंगे? तो जनरली ईडीटीए बोल के कोई भी चीलेटर, डीएमपीएस और डीएमएसए, दिस टाइप ऑफ सम chelators components are there which has the capacity to chelate that bivalent cations or trivalent even to some extent. So we people give it, drink कर लिया तो फिर से पिला देते हैं. That is also a toxic component, but that attached with your bivalent component and without reaction through the outsource, मतलब gastrointestinal tract, it will goes off from the system. So you have no concern about that. And these are the phenomenon of a very common, some of the common essential metals का recovery के लिए और acute poisoning से recovery के लिए use किया जाता है. Zinc, manganese, selenium, iron can be provided and protected as because it has that capacity to substitute cadmium from the system. But all those things in a higher amount which you are going to give to substitute cadmium which is already present in the system in a higher concentration, that is also a very problematic component. So that is one thing. And vitamin scavenging is the best choice. So vitamin E has been generally utilized any of the heavy metal contamination, first charging the vitamin E, D, these type of vitamins. A, B, C is not so easy because they are absorbed by themselves very quickly. So if they absorb, they may take some of the components inside. So generally D or E after दिया जाता है. Generally E दिया जाता है first. So to overcome the cadmium toxicity. Probiotics means some of the bacterial complex, bacterial दही lactobacillus मैं बोल सकती हूँ, but ऐसे कोई components is also can be utilized and generally people utilized. दही खाने से आपका कुछ भी ऐसे contamination, minimum amount of contamination can be waste out from the system. And other than that, plenty of phytochemicals are nowadays utilized. One of them is also utilized. We have started to utilize and see in our lab also. So these are the components are very commonly known to overcome the oxidative damage. If they can overcome the oxidative damage, then it is a partial or full total substitution of all the oxidative damage generated by the cadmium chloride. So it can be, I am not telling it must be, but they have that capacity, but how much they have and how they can do it. So that is a criteria of analysis. And so phytochemicals of those components are very common. So now I am going to some of the specific data, but I will shortcut it as much as possible. Here is one paper of us, brain is, actually मैं इस photographs में दिखाया कार्डियोवास्कुलर सिस्टम दिखाया, लंग्स दिखाया, पल्मोनरी डिजीज है वो दिखाया, किडनी दिखाया, लीवर दिखाया, इंटेस्टाइन दिखाए, इवन बोन्स भी दिखाए. बट मैं किसी जगह में ब्रेन को आई वोंट शो एनीथिंग हियर, बट आवर डाटा शोज दैट इट इज नॉट दैट कि only these are the uncomfortable organs, because we detect it very quickly. But it has been found in brain, the composite activity is much more higher. So here I am giving some of the gist of those data. We utilize cadmium chloride on Swiss albino mice model. The models are, if the models are changed, then the activity also change. So it has also been found by us that the even mice of different breed can affect in a higher or lower concentration. So in case of, we have found the Swiss albino mice is the most stubborn. They are very less activity, and we have taken that. And we utilize only, you can visualize only 2
So we have a short technical glitch. Elora ma'am. Indeed, a very important and interesting and engaging discussion over here for cadmium toxicity.
ma'am is back,
Yes,
ma'am
ma'am,
is
we
back.
lost your connection, ma'am, for a certain time. Elora ma'am.
Ma'am, please unmute yourself. Here she is.
So now I can again join.
Ma'am, you it's fine now. You may please share. We might have lost you for a minute only.
So my slides are visualized now? No.
No, ma'am. You'll need to share them again.
Okay. Just. So now it is okay?
Ma'am,
Yes.
it's coming on the screen. Yes, ma'am. It's fine.
So now I will start again from some back things, that 2 mg per liter we are showing here for a mice model, and for mice model of that age and that Swiss albino, it is 22 mg per liter is the criteria of its permission level. So you can understand, we are taking a very lesser amount, not above the amount of its toxic level. So on that we have, on that state also, on at that status also, we can found a huge amount of loss of at least for 60% loss of mice, Swiss albino mice. So if you persistently, continuous 8 days, we are giving this feeding of cadmium chloride, and after 15 days we are going for 8 plus 15 days, and it's a matured mice. So when we are going for them around another month or so, we are found that huge amount of ROS has been identified in their serum, as well as kidney, liver, spleen, testes, and brain. Fortunately, unfortunately, my student has decided to go for brain. As I have claimed that there will be no change because no report has been there. So please don't go with the brain, because brain handling is very tough. But even after that, he goes for that, and found interestingly, brain also shows some damage. And you understand probably that kidney, liver, spleen, testes may have liver is in our system, it is around 5 kgs of component in a adult body, 5 to 7 kgs maybe. And in kidney, one may lose activity to another can substitute. A spleen is okay, you maybe have a, but test is also in such a, that way. But in case of brain, if one point has been inactivated, then you may lose one activity of your lifetime, for your lifetime. So, but we have found that only 2 mg per liter concentration of water, if has been feeded continuously to a adult rat for 8 days, and after 15 days, the 60% there will die. So our concentration level is higher than LD50. Though 25 to 30 is the level of its permission of cadmium toxicity. But that toxicity is not that every day you are pilau. I accept it. But if these 8 days may shift to 8 months, then it also happen, because cadmium ka degradation process is very less. It's a heavy metal compound. So you understand, though there is a facilities we are providing, time loss ho gaya. I'm sorry. So I am giving the informations only that brain, when we go for brain sectioning, and BAX is a component which has to be identified for apoptotic death or induction of death. So it is also high in brain. Expression of brain is much more higher than any other organs. So you understand, other places there is a capacity. In case of serum DNA leke, cell leke, serum se cell leke, when we identified, we have found a huge amount of DNA damage. And when we correlate with some of the or biochemical processes like SOD, catalase, glutathione, LDH, and LDH is lactate dehydrogenase, and LPO is lactate peroxidase. And lactate peroxidase is much more concerned with lipid peroxidase, is much more concerned enzymatic component in case of any neural system, that is brain. And it has been found that is much more variable in all the identification process we have gone through by plenty of correlation analysis. And is when it has been found that LOP, you can see here, brain is a must to identify with LPO, and all other systems are also. So LPO is a good identifying biomarker by which you can sense that its neural abnormalities are going to start. So brain is very much susceptible for that. We have found another very good identification we have found from the liver itself. We utilize the streptozotocin induced diabetic mice, and we fed with the cadmium to our mice, and we have found in both the cases there are plenty of biochemical abnormalities have found expressed, and which can give plenty of abnormal conditions, abnormal expression of proteins which are correlating with diabetes, hyperglycemia. In a sense diabetes कहते हैं. So all diabetic proteomic analysis has been found that those are the components which are very crucial. Carbohydrate metabolism has been totally disturbed. 55, score level is 55 is very, very high. 2, 3, 4 is okay, but 55 is very high. And it has been found that carbohydrate metabolism, nucleic acid metabolism, as well as small molecular biochemistry, very, very small molecules of proteins has been destructed. So the basic signaling pathway, just P53 has been disturbed, we have found. These type of very basic components are very much disturbed with cadmium chloride toxicity, as well as carbohydrate metabolism, which gives us a very good signal that metabolic conditions has been also disturbed by cadmium toxicity. So I won't go for these. And lastly, we also indulge another problem, complicated problem, that it has been found with cadmium toxicity, which is not found by many of the bivalent heavy metal toxicity. That is, one of them is cadmium, which shows the gut microbiota disturbances. It has been already proposed and found that there is a 2012 and 2015, it has been found that there is a disturbed microbiota in gut system. But now we are interested to see how we can control it and disturb the after disturbance with cadmium chloride, whether we can control it. So it is not that toxicity is the problem. But in our advanced world, we also must think about how we can overcome it. There is a toxicity. We cannot absorb all the electronic components, waste components within our system. But there must be some circulation of cadmium in the solution or water or drinking water. But we must have to think about some of the remedies. And for that, we are in our lab nowadays, we are working on some of the phytochems, which we have shown some capacity to overcome that cadmium toxicity, but it is under publication, so I am not going to discuss about it. Here is Sandeep and Komal, these two students. Sandeep is already done PhD, go abroad, and Komal is now working on gut microbiota. So this is the things. Thanks for the this thing, for your kind attention. Thank you very much. I am sorry I am late by 5-10 minutes probably.
No problem, ma'am. Thank you so much. And it was a deeply engaging and a very interesting presentation on the cadmium toxicity, the prospects, and the future aspects even of bioremediation, as you said. So these are one of the very important topics that are all participants would have gained an insight from. Thank you so much, ma'am. Ma'am, there are a few questions which I'll like to read very fast, as we are left with only 2 minutes now. So can a healthy dietary lifestyle prevent the effects of cadmium toxicity on body? This question is asked by Dr. Juhi Gupta. Ma'am, may I ask you to unmute, please?
Oh, sorry. By 100% Juhi, actually, zinc, iron, I have to tell some of them, some of the components which are present in green vegetables, but along with that, those green vegetables contain high amount of cadmium too. So there must be a balancing, but yes, of course. So shortly, yes, of course.
Okay.
Thank you, ma'am. And the other question is by Shivangini. So she's asking, can cadmium also associate with other heavy metals causing lethality?
Here I can say something that is a synergistic effect of cadmium. Probably she is asking about that. Some of the work has been done, not by me, but some of the work has been done, and it has been found if it is with arsenic 3 or some other 5-3 component, then only cadmium helps. But if it is with plus plus to any component, iron is much more active, cadmium is very less active. So even arsenic or maybe manganese, magnesium, these type of components cannot, selenium, they cannot, cadmium cannot overcome their activities. So synergistically, if you think, other bivalent cations are more active, and for that reason till 1996 97 only restricted, cadmium toxicity has been restricted to lungs, and all people are worked on lungs and smoking intake of cadmium. Never, people can never think even ki cadmium can come within the system other source than inhalation. So because the absorption is very less, when I am comparing with iron or any other sodium plus 2 component, cadmium is the very less effective component to be absorbed by the system.
Thank you so much, ma'am, for this beautiful overview of cadmium toxicity, for the systemic toxicity, just not focusing on the various aspects of toxicity, but also giving us a hint of how we can resolve the problem. And there are various measures that are required to limit the use of cadmium also, so that the toxicity can be prevented. Thank you so much, ma'am, for joining us today. We are highly honored to have you here.
Thank you very much. Thank you. May I leave now? If
Yes, ma'am.
any queries from any of the students, can mail them also, maybe forwarded as well as maybe send it to me. Okay, thank you very much. Thank you.
Sure, ma'am, please. We will surely put up all the queries that come up to you, ma'am, so that they can be resolved within the time. Thank you so much, ma'am.
Thanks a lot.
So after our brilliant discussion with Dr. Elora Ghosh, we will now move on to the next speaker, that is Dr. Rajesh Kumar Sharma. He has joined us. He is Assistant Professor, Laboratory of Ecotoxicology, Department of Botany, Banaras Hindu University. He's also former scientist, GBP, NIHE, MOEF and CC, Government of India, Almora. His research area is ecotoxicology, with an experience spanning 2 decades and having H index of 22. He has many research publications and awards to his credit. Sir, we welcome you, and we would like to honor you with our traditional online virtual sapling. I request Mr. Naresh to please showcase the Tulsi sapling. Dr. Amrina Sardar Khan, Assistant Professor AIS, is offering the virtual Tulsi sapling to Dr. Rajesh Kumar Sharma, sir. Sharma, sir.
Sir is sharing his screen. Welcome, sir.
Yes, the things are going on excellently. So now I also add on to for the invitation to Dr. Rajesh Kumar Sharma to deliver on risk assessment and sustainable management of heavy metal contamination. And you had a wonderful discussion and presentation earlier on focused on cadmium. So I think if we merge these two and see from the toxicological perspective, it would be beneficial and also gives the way what we can do in the future. What are the gaps? How we can identify? And now the era has come when we have to see that it is not the problem, but the solution base. So I would like to interfere at this moment and also give you a point that every one of us are looking for solution based and how we can merge to these two. I think my other speaker will be also focusing and telling us the pathway for that. Thank you. So now we can have the continuation of the activity, please.
Thank you. Thank you, Sharma, sir. It is always delightful to have your input, sir. Dr. Rajesh Kumar Sharma, sir.
कम है ना ये इसका क्या था
Sir, we can see your slides, but sir, your camera.
Sir, please
Sir, I
unmute
request
yourself.
you to
Rajesh,
please
sir.
unmute. Sir, please unmute, Rajesh, sir. Yes, sir. Now it is unmute.
Okay, okay. Now I am audible and also visible.
Yes, sir. Absolutely.
Okay. My
Yes, sir.
slides are visible or not?
Yes, sir. They are visible, but can you put them on slideshow mode?
Yeah, sure. Yeah. Is it okay?
Perfect,
Uh,
sir.
Sir, yes. Thank you, sir.
Okay. Just a minute. So, very good very good morning to all the participants and the organizer. First of all, I would like to thank the organizer and especially Dr. Asutosh Tripathi for inviting me to deliver the talk in the ongoing workshop. So I will speak on the risk assessment and sustainable management of heavy metal contamination in Indo-Gangetic plains of India. So before this talk, one talk was delivered by Madam Ilora Ghosh. She has talked much about the cadmium toxicity, that is impact of cadmium on human health. So I am just going to give you a picture about the cadmium, about the heavy metal, including the cadmium in the ecosystem, particularly the agriculture ecosystem, that is in soil, water, and the vegetable crops. In addition to that, I will also talk about the possible health risk to the local people of the studied area. Participants, you all know the importance of soil, and I am here telling you the importance of soil in terms of a quotation, that is, the nation that destroy its soil destroy itself. And this was given by the Franklin D. Roosevelt. So a nation, a healthy nation, is depend on the soil. If the soil is healthy, the nation will also be healthy. Before starting the talk, I would like to give a brief introduction about the area where I am working. So particularly, I am working in the field of ecotoxicology, where I am monitoring the level of heavy metal in different metrics of the environment, that is soil, water, and the agricultural crops, and then assessing the possible risk, health risk, by using some health indices. And then I am also trying to find out the solution for the heavy metal contamination, and that is the phytoremediation is one of them. In addition to phytoremediation, we are also trying to use some organic amendment to reduce the contamination in the soil. That will also reduce the contamination in the agricultural crop, that is edible part, and consequently in the food chain. So this is the team, research team, who are helping me in doing this type of work. So here, this Mr. Prince Kumar Singh, who is working on wastewater risk assessment, and I will give you the work what he has done during last 3 years. In addition to his work, I will also give a brief account of the work of Umesh Kumar and Jaishankar Yadav, who are using the agriculture soil amendments and the microbial consortia for the remediation of the heavy metal contaminated soil. Is it okay?
Yes, sir.
Okay. Here I am just telling you about the soil pollutants. So if we are talking about the soil pollutants, so there are 3 types of soil pollutants. One is the mineral oils, which account 20%, hydrocarbon, which accounts 42%, and the heavy metal accounts 31%. And other soil pollutants, which accounts only the 7%. According to the report of Ministry of Environment, Forest and Climate Change 2015, there are some sites which are identified as a heavy metal contaminated site. So here I have just given you the list of the state where the number of sites are recognized the contaminated site. For example, in Uttar Pradesh, 40 sites are identified as a heavy metal contaminated site. This is one information. And in terms of lead exposure, if we see the lead contamination or lead exposure, so among 180 countries on the globe, India ranked 174. That is environmental performance index is 21. So in terms of lead contamination, the India ranked at 174, where other metals are not included. In earlier presentation, you have been told about the effect of cadmium on various tissues. That may be spleen, liver tissue, or kidney tissue. Why we are considering the heavy metal only as an important soil pollutant? According to V.I. Warnadisky, the influence of a contaminated soil on the ecosystem is directly dependent on the metal compound in the soil. So this heavy metal is important because the whatever influence of a contaminated soil is observed on the ecosystem, which is directly dependent on the concentration of metal in the soil. We can define the metal as heavy metal as metal which have a specific density more than 5 grams per centimeter cube. It means these heavy metals are heavier than the other metal in terms of the density. Globally, there are 5 million sites of soil pollution covering 500 million hectares of the land, in which the soil are contaminated by different metals or metalloids with present soil concentration higher than geo baseline or regulatory levels. So this report also confirmed that the sites, there are many sites on the globe which are exceeding the geo baseline or regulatory level. So if we are talking about the impact of these heavy metals on the plant system, they are affecting particularly the yield of the plants or agricultural crops, as well as pose risk to the safety of the food crop or agricultural crop by getting accumulation more than the permissible limit in the edible parts. So these heavy metals are present in the soil. They are taken up by the roots of the plant and then transported to the different parts of the plant, maybe stem, maybe leaf, or edible part of the plant. And they are reducing the yield of the agricultural crop or plant by affecting the physiological and biochemical activities. These heavy metals can reduce the photosynthesis, the respiration, chlorophyll content, protein synthesis, as well as the overproduction of the reactive oxygen species, alteration in the metabolic activities, osmotic balance, damage to lipids and the DNA, cell injury, and ultimately the death. The uptake of these heavy metals by the plants are depend on many factors. For example, the plant species, the properties of the medium, root zone, environmental conditions, chemical properties of the contaminants, bioavailability of the metal, chelating agent added. The accumulation or the uptake of these heavy metals depend on the plant species, because one plant species can accumulate more heavy metal as compared to other plant species. For example, leafy vegetables accumulate more heavy metal as compared to the root vegetables. Similarly, the properties of the medium, for example, soil is the medium, growth medium for the plant. The pH and the organic matter content in the soil will affect the availability of the metal as well as the uptake of the metal by the plant. Now, the sources of the heavy metals contamination. So sources are categorized into the two groups. One is the natural source and another one is the anthropogenic source. Natural source includes volcanic eruption, forest fire, fly ash, aerosol formation, weathering of rocks and the minerals. Whereas the anthropogenic sources includes the intensive agriculture and the forestry, where fertilizers and the pesticides are added to the soil. Wastewater is used for the irrigation. The industrial activities, which includes the mining industries, fossil fuel combustion, smelting, coal mine industries, metallurgical industries, and some other miscellaneous sources. For example, water treatment, used battery, medical waste, vehicular activities, domestic waste, sewage sludge, and electronic waste. So these sources are adding the heavy metal to the soil. If you see this diagram, so here you can see the heavy metals, which are the contaminants. When these heavy metals are added to the soil through various activities or from various sources, so these heavy metals are getting accumulated into the different parts of the plant through the root uptake. Then these heavy metals which are accumulated in the edible parts are going to the food chain, and food chain means the when we are consuming these contaminated vegetable, particularly the edible part, we are getting exposed to the heavy metals. Risk assessment of the heavy metal contamination. The soil soil contamination become a major threat to the food safety and the security on a global level. As I told you that these heavy metals are getting accumulated in the edible parts of the food crop, and they are posing safety risk to the food safety. This may be at a local level or national level or global level. Several soil contaminant issues, for example, movement of the contaminants through the soil, effect of soil contamination on ecosystem health, effect of soil contamination on food safety, soil contamination type, for example, microbial, heavy metal, chemical, radiation, and toxic, and the impact of modern agriculture on the soil contamination and the quality. Several ideas proposed for assessing the food safety and the security problem, which will be essential for sustainability, meeting the current and future demand for the food supplies. So I have told you about the sources of heavy metals, how these heavy metals are moving in the ecosystem, that is from source to the soil to the crop during their growth, and then to the kitchen, that is available for the consumption, and finally it is getting accumulated in the human body. So we have various indices which can be used or which are commonly used for the risk assessment of the contaminated soil. So I am going to talk about some indices which are used for the risk assessment of the contaminated soil. The first one is the metal pollution index. Metal pollution index indicates the relationship between the contaminated soil and in reference soil. That is the ratio of heavy metal concentration in the contaminated soil to that of the reference soil. And we are getting some values which may be less than 1 or maybe more than 1. So based on that value, we can classify the soil in different category. For example, very slight contamination. If the MPI value is less than 0.1, slight contamination if the values are in between 0.1 to 0.25, and if the value of MPI is more than 16, then we can say the excessive pollution or excessive concentration of the metal in the soil. So this is based on the MPI, that is metal pollution index. Then second index is degree of contamination, which denotes the sum of MPI for all the heavy metals at a particular contaminated site. So this MPI is calculated for individual heavy metal, and when we are adding the MPI value of all the studied heavy metal, that will give the degree of contamination. And based on degree of contamination, we can also classify the soil as low degree of contamination, moderate degree of contamination, considerable degree of contamination, very high degree of contamination. That is, if the C DEG is less than 8, then we can say the low degree of heavy metal contamination. If the value C DEG value is more than 32, then we can say very high degree of contamination. Is it okay?
Yes, sir.
Okay. Then the third indices is pollution load index, which is the geometric mean of the MPI of all the heavy metals in the contaminated soil. If PLI is 1, then we will say that there is no pollution, and if it is in between 1 to 2, then it is moderate pollution. If it is in between 2 to 3, heavy pollution, and if it is more than 3, then it is extremely heavy pollution. Then the fourth one is the ecological risk factor, and fifth one is the potential ecological risk factor. Ecological risk factor used for the expression of the ecological risk for a particular heavy metal in contaminated soil, and potential ecological risk factor indicates summation of the multiple heavy metal determined in the studied contaminated soil. So based on this value of ER and RI, we can also classify the soil in different category. If here you can see the ER value, if it is less than 40, then there is low ecological risk, and if the value of RI is less than 150, then it is low ecological risk. So both are comparable, because one index is based on individual metal, that is ER, and other index, that is potential ecological risk factor, which is based on the summation of all the heavy metal studied. This is related with the soil risk assessment of heavy metal contaminated soil. Now I am coming for the vegetables or food crop. So there, this table is showing the safety limit of the heavy metal concentration in soil, irrigation water, and the plants. So here you can see the Indian standard, WHO and FAO, European Union standard. So three safety limits set by different government or the regulatory body to control the heavy metal in the soil or in the water or in the plant or food crop. So in case of water, that is irrigation water, here you can see, according to the Indian standard and the FAO, the permissible limit or safety limit is 0.01 mg per liter or microgram per liter. Similarly, in case of food crop, you can see here the cadmium permissible limit or cadmium safety limit, according to the Indian standard, is 1.5. Whereas according to the WHO and European Union regulation, it is 0.2. So we have to maintain, or we have to regulate the metal concentration within this permissible limit for the safe society. We can, based on the heavy metal concentration in edible parts of different vegetables, we can calculate some health risk indices for the heavy metal. This one is the enrichment factor. Enrichment factor is the mobility of heavy metal in soil plant system under different irrigation regime. That is the accumulation of heavy metal in crop produce under wastewater irrigation over the groundwater. So this is the ratio of heavy metal concentration in the vegetable crop divided by the heavy metal concentration in the soil. at the contaminated site, and heavy metal in vegetables divided by heavy metal in soil at the reference site. Okay, so this is the ratio of the VW by SW to at the contaminated site with VG and divided by SG at the reference site. The bio concentration factor and the transfer factor. Bio concentration factor is was calculated by dividing the heavy metal concentration in the root with that of in the soil. That is heavy metal concentration in root with heavy metal concentration in the soil. The transfer factor, which is calculated by dividing the heavy metal concentration in edible part by dividing the heavy metal concentration in the soil. So this bio concentration factor and the transfer factors can also be used for the health risk assessment. Here the metal pollution index, that is the geometric mean of concentration of all the heavy metals in samples. N is denoting the number of heavy metal. So MPI is also calculated or computed for the health risk assessment of the heavy metal contaminated vegetable crops. Then daily intake of heavy metals, that is the daily intake of metal by the local residents of the study area by a consumption of contaminated vegetables and cereal crop was calculated by using this formula, that is DIM is equal to M multiplied by K multiplied by I divided by body weight. So M is the concentration of heavy metal in vegetable or cereal crop. K is the conversion factor, that is which is used for the conversion of fresh weight of sample to their dry weight. And I is the daily intake of vegetable and cereal crop by the children or the adult people, and BW, that is the average body weight of the children and the adult. So by using this formula, we can calculate the daily intake of the metal. Then hazard quotient. Hazard quotient is calculated by multiplying this daily metal intake with the exposure frequency and the exposure duration divided by the mean exposure time and the reference oral dose. So this hazard quotient is also giving an idea about the exposure of the human to the heavy metal contamination through the dietary intake of the contaminated vegetables or food crop. Now I am going to give you the result of one study which will tell you the status of the contamination at a particular site, that is that site is located in the suburban areas of the carpet city, that is Bhadoi. So Bhadoi is popular for the carpet industries. So these are the industries which are located in the studies area, and these blue line is showing the drain. The length of the drain is approximately 10 kilometers, and we have selected 4 sites: S1, S2, S3, and S4. So these are the agricultural area where the wastewater is used for the irrigation. And since the last 10 to 15 years, and this wastewater is discharged to a local river, that is Morwa River, and through this river this water is coming to the Varna River, and then it goes to the Ganga River. We have also selected one site, that is the reference site, which is located 5 kilometers away from this agricultural area where wastewater is used. So this reference site particularly we select to compare the values of the contamination of the vegetable, soil, and the water. So this is the carpet. Sorry. This here I am trying to show you the situation or the agricultural area. This drain is showing the carpet effluent, and this drain is showing the domestic effluent, and this both effluents are mixed here and then goes through the this open drain to the Morwa River, and to this drain the effluents from other industries are also coming. This is the vegetable which we have collected during the pre-monsoon season, particularly the vegetable crops, and this is the wheat crop which we have. This is the rice crop which we have collected during the monsoon season, and this is the wheat crop which we have collected during the post-monsoon season, along with the soil as well as the water sample. So if we see the chemical characteristic of the wastewater as well as the groundwater, which are used for the irrigation of the agricultural crop at the wastewater irrigated area as well as the reference site. So here you can see the value of all the heavy metals are within the permissible limit set by the WHO, except the cadmium. So here you can see the cadmium concentration at site 1 and at site 2 is more than the permissible limit, whereas site 3 and site 4 it is less than the permissible limit. Overall, the mean value is also above the permissible limit, that is 0.01. So cadmium is problem here in terms of the wastewater. Wastewater is containing the cadmium more than the permissible limit. Now here we can see the seasonal variation in the concentration of studied heavy metal or tested heavy metal. So in groundwater, the cadmium and chromium were not detected, whereas other heavy metals were present in the groundwater. In wastewater, all the 5 heavy metals has been detected during all the season, that is pre-monsoon season, post-monsoon season, and the monsoon season. So this figure is showing that there is variation in the concentration of heavy metal with the season. If you see the concentration of the cadmium concentration was found maximum, that is 0.012 during the post-monsoon season. Okay, and in monsoon season and pre-monsoon season there is no change in the concentration of cadmium. I am focusing on cadmium because cadmium is more than the permissible limit. Now this is table is showing the concentration of different heavy metal in the soil. All the heavy metals are within the permissible limit, except the cadmium and the nickel, which is near to the permissible limit, that is 3 in case of the cadmium. 3 is the permissible limit, and 2.35 is the concentration of cadmium in the soil, and in case of nickel it is 50, and the detected concentration of nickel is 36.30. So this is this slide is also showing seasonal change in the concentration of tested heavy metal in groundwater and the wastewater irrigated soil. Now the important thing is the availability of the metals. So we have analyzed the 5 fractions of the heavy metal in the contaminated soil and the reference soil. That is F1, F2, F3, F4 and the F5. The F1 is the extensible form of the heavy metal. F2 is the carbonate bound form of the heavy metals. F3 manganese iron oxide bound heavy metal. F4 organic matter bound, and F5 is the residual fraction. F5 is the concentration of the metal which is not available for the uptake of plant, whereas this F1, F2, F3, F4 are available for the uptake of the metal. This multi-level donut graph represent the effect of seasonal variation in different on different fraction of the copper and zinc in both the groundwater and the wastewater irrigated soil. So if we see the cadmium concentration, different fraction of the cadmium in the wastewater irrigated soil, here you can see the innermost circle is showing the pre-monsoon, then monsoon and the post-monsoon. The percent of the fraction of different fraction of cadmium is changing, okay, during the different season. So F1, this F5, that one is the residual form. The residual form is maximum during the pre-monsoon season, that is 41.2%, followed by the monsoon season, that is 28.3%, and list in the post-monsoon season, that is 35.1%. So all the 5 fraction of each heavy metal at both the sites are varying with the seasons. Now let us come to the metal pollution index and the degree of contamination. I have already told you how these indices were calculated. So this metal pollution index was calculated for each metal during all the season. Here you can see the nickel. The nickel index, metal pollution index for the nickel is maximum in the pre-monsoon season, and the values are ranging in between 1 to 4. So based on this MPI value, the soil is slightly polluted as well as moderately polluted in terms of the particular heavy metal. If we are observing the cadmium contamination, then we can say it is slightly polluted with the cadmium, and when we are observing the MPI value of nickel, then it is moderately polluted. And in terms of degree of contamination, the all the values are in between 8.1 to 16. So the tested soil was found moderately polluted with all the heavy metals. Now the pollution load index, was found more than 3. Based on this index, the soil is extremely polluted with the heavy metals. Here you can also see at the contaminated site, the pollution load is very high as compared to the reference site during all the studied season. The ecological risk factor, the value of ecological risk factor is more is more than 40, that is moderate ecological risk in case of the cadmium, and in case of other heavy metal, that is chromium, nickel, copper, and zinc, it is less than 40. And if we are observing the value of the potential ecological risk factor, according to this index, the contaminated soil is posing low ecological risk to the ecosystem or to the suburban ecosystem during all the season. Okay. So because here you can see the value is less than 150. Now I am coming to the concentration of metal in different vegetable crop. This one is for the cadmium. So this pie diagram is showing the concentration of the metal, cadmium metal, in edible part and non-edible part. So if we see the pie diagram for the palak and the cabbage, so cadmium is more accumulated in the edible part in case of palak and the cabbage as compared to the non-edible part. Whereas in other tested vegetable, it is vice versa. This bar diagram is showing the concentration of cadmium in different vegetable at different site. So the cadmium concentration in all the vegetable and at all the site, including the reference site as well as the wastewater irrigated site, the concentration is more than the permissible limit of the WHO and the FAO standard. Whereas if we compare this value with the PFA, that is Indian standard, it is more than the permissible limit at the wastewater irrigated site, whereas within the permissible limit at the reference site. This is for the chromium. So chromium is also found higher in the edible part in palak and the cabbage, whereas in other vegetable it was higher in the non-edible part. The chromium concentration at different site was shown here. If we compare this chromium concentration with the Indian standard, this site, the chromium concentration in edible part of wheat exceeded at site 1. S1, the chromium concentration in wheat is exceeding the Indian permissible limit. In case of nickel, the nickel concentration is also exceeding the FAO and WHO permissible limit, as well as the Indian standard at most of the site in most of the vegetables. Copper was found within the permissible limit except in wheat at the site S1. Zinc is also exceeding the permissible limit of both standard, that is Indian standard and WHO standard at some sites in case of some vegetable, for example palak, radish, and the brinjal. Now this table is showing the mean value of the enrichment factor and bioconcentration factor of the heavy metal for the different food stuff collected from the suburban agricultural areas of Bhadoi. The enrichment factor and the BCF factor was found more than 1 in case of the vegetables, in case of all the vegetable in for zinc, nickel, copper, cadmium, and the chromium. But in the bioconcentration factor was exceeding exceeding the unit value in case of cadmium for all the vegetables. When we see the mean value of zinc was more than 1 in case of more than 1 for all the vegetables, because this is the mean of the all the vegetables. Now this is the transfer factor of different heavy metals at the contaminated site. For all the heavy metals, the transfer factor was found below the 1, below the unit value, except the cadmium. So the transfer factor for cadmium was more than 1. It means this the accumulation of cadmium in edible part of the tested vegetable was more than their concentration in the vegetables in the soil. Okay, so if we see the order, the transfer factor of cadmium was found maximum, followed by zinc, then nickel, then copper, and least for the chromium. Here I am showing you the metal pollution index of the groundwater and the wastewater irrigated vegetables. Okay. So here you can see this blue dot is showing the metal pollution index for the vegetables collected from the groundwater irrigated area, and this red one is showing the metal pollution index for the vegetables and the cereal crop collected from the wastewater irrigated area. This value is metal pollution index value for the vegetables and crop from wastewater irrigated area is ranging between 9.05 to 18.34 with a mean of 12.87. So this metal pollution index was maximum for the wheat crop or wheat grain, followed by this paddy, and then the cabbage. So this is the daily intake of heavy metals and the hazard quotient of different heavy metals via the dietary intake of tested vegetables. So here the daily intake of the metals through daily intake of metal by the consumption of all these vegetables was found low, that is less than 1 in case of all the vegetables. If we see this value it is almost either higher or equal to the children, means children are taking more, children are exposing to heavy metals more as compared to the adults. If we compare the value of DIM here in case of Oryza sativa, that is rice grain, so this value is higher for the children, that is 0.164, and in case of adult it is 0.143. The hazard quotient for of all the heavy metals in all the vegetables were found less than 1, except the cadmium. If you see the HQ value of cadmium for all the vegetables, it is more than 1, and this value is also higher for the cereal crop, that is rice and the wheat, as compared to the vegetable crops. So this is the status of heavy metal contamination in wastewater irrigated area of the suburban wastewater irrigated area of the Bhadoi. Now, what are the approaches for the remediation of this contaminated site? So as we know that there are three methods for the remediation. One is the physical method, chemical method, and the biological method. So we are particularly focusing on the biological method because it is environmental friendly and the cost effective, whereas the physical method and the chemical methods are very expensive, are having high cost. So phytoremediation is mostly concerned, that is remediation of the contaminated soil or contaminated site by using the plants. So there are some methods, that is phytodegradation, phytovolatilization, phytostimulation. These are the method, or phytoextraction, that is accumulation of the metal in the edible parts. So if we are talking about the phytodegradation, that is use of plant to destroy the organic pollutant. Phytovolatilization, that is conversion of metals or metalloid to the less toxic and the volatile form. And when we are talking about the phytostimulation, that is degradation of the organic pollutant by plant root exudates. And phytoextraction, that is metal chelation or metal solubilization. So the utilization of plant to remediate toxic chemicals found in the contaminated soil, groundwater, sediments, wastewater, and the surface water. So we can use the plants for the remediation of metals in soil, water. These are the target metal, that is cadmium, chromium, arsenic, lead, selenium, copper, zinc, barium, nickel. And these plants can be used, that is sunflower and mustard forming, Chinese sprague fern, and southern cattle combined.
Sir, may I humbly intervene?
Yeah.
So we are 10 minutes over time for your session.
Yeah, I am just going to show what are the remediation approach in 5 minutes only.
Thank you, sir.
Okay. So here we can use the plants for the remediation of contaminated soil. We can use the biochar. We can use the rice. We can use the ash, that is ash produced from the organic biomass, and the microbes. So I am just going to show you some results. We have screened 5 varieties of the mustard to identify the variety which is accumulating lower concentration of the metal. So out of 5 varieties of the mustard, we identify the NRC variety of the mustard is accumulating lower concentration of the metal at both the site, that is the wastewater irrigated site and the groundwater irrigated site. In terms of the heavy metal load, this NRC is having a low heavy metal load as compared to the other varieties at both the site. We have used the rice husk biochar. We have used different concentration of the rice husk biochar, that is 1%, 2%, and the 4%. And at 2%, we are getting the interesting result. That is 2% application of 2% rice husk biochar to the contaminated soil reduce the cadmium, chromium, nickel, zinc, and the copper significantly as compared to the control, that is contaminated soil. Then we have used the ash which was produced from the sugarcane bagasse and the rice husk for the remediation of nickel contaminated soil. So here the nickel accumulation in the edible part of seed is shown in 2 varieties of the wheat and 2 variety of the rice. So you can see the 4% application of both, that is rice husk ash and sugar bagasse ash, reduce the nickel concentration significantly in the seeds of the tested plant species, that is wheat and the rice. We have developed a consortium of the microbes, that is Eco 1, Eco 2, Eco 3, Eco 4 and Eco 5, which contain 2 microbes or more than 2 microbes. This Eco 5 is the mixture of 5 microbes, and Eco 1, Eco 2, Eco 3, Eco 4 are the mixture of 2 microbes. So here the mixture is accumulating more heavy metal as compared to the individual microbe. And the Eco 4, which is having 2 microbes accumulating more heavy metals, particularly the cadmium, as compared to the other consortium. We have applied this consortium to the contaminated soil, that is naturally contaminated soil, that is wastewater irrigated soil, and we observe the response of the tested crop in terms of test weight, and the harvest index. All the microbial consortia improve that test weight and the harvest index of the tested plant species significantly, whereas Eco 4 was found the prominent one among the tested microbial consortia. When we observe the value of different heavy metals, that is copper, chromium, zinc, nickel and the cadmium in both the tested species, that is mustard and the wheat, this Eco 4 was found very effective in reducing the concentration of the tested heavy metal in the edible part of these 2 plant species. Now, based on my result, I can suggest that vegetables or crop grown in wastewater irrigated areas should be monitored regularly for heavy metal contamination to ensure the safety of local people. Different varieties or cultivars of food crops should be screened to identify the low accumulating crop for the cultivation in the contaminated area. In the last, the potential application of microbial consortia, biochar and the bioash as an amendment should be explored to reduce heavy metal accumulation in crop and consequently in the food chain. In the last, I would like to acknowledge the Head Department of Botany, Banaras Hindu University, for providing the basic facility, research facility, and the funding agency, that is BHU, CSIR, Institute of Eminence, SERV, and UGC for providing the financial support to the research. Thank you very much.
Thank you, sir. Thank you for addressing our participants and enhancing their knowledge as regards to heavy metal contamination and bioremediation techniques. Thank you so much, sir, for addressing us today. So can we take just one quick question, because we are really short of time. The other questions, we'd like you to answer it on a personal basis later on if possible.
Sure.
So do we use criteria pollutants values in deciding the pollution index scale? That's the question by Dr. Juhi Gupta.
Can you repeat the question?
Do we use criteria pollutants values in deciding the pollution index scale?
There are some indices. There are some index where we are using this value. not in all, but there are some index. For example, cancer risk assessment, where we are using this criteria value.
Okay,
And one
thank
I
you,
have-
sir.
One I have shown that is hazard quotient. There also we
Yes.
are using some values.
Absolutely, sir. Thank you. The other question is, sir, can you suggest some easy morphological characteristics that may help to differentiate heavy metal contaminated vegetables for a layman?
Morphological characteristic.
Yes, sir.
We can't suggest, we can't recommend any morphological characteristic
That's true.
which can directly be used
That's
for the
true.
assessing the
Sir, yes. Another question is by Dr. Kartikeya Shukla, why the heavy metals are treated as the most beloved target for remediation, even though traces of them are highly beneficial?
The heavy metals are categorized into two groups. One is the essential and other one is the non-essential. Non-essential heavy metal, for example, cadmium. This cadmium is toxic to the human being or to the biota, even at very low concentration. And they are non-degradable in nature. That's why there is need for the remediation of these heavy metals. And for the essential heavy metal, for example, copper and zinc, since they are essential, there is no problem with them. But if their concentration is very high, in that case, there is need of the remediation. For example, zinc smelter industries. So zinc concentration is very high in the vicinity of such industries. Are you getting my point? Hello.
Thank you so much, sir. And we are very thankful for you to answer the question so quickly. There are other queries, but we'd like to send them to you.
Richa, VP sir has raised his hand. I think he has something to say.
VP Sharma, sir,
Yes.
would you
have
like to
just-
say something, sir?
Good afternoon to each one of you. I would like to have the information regarding, this is the International Year of Millets. So
Yes.
whenever BHU expert is there, I would like him to say a few words that how the International Year of Millets and what can be done in the field of the botanical developments, consumption, hunger point of view, in linking up with the SDG point of view. Because I have not heard his complete lecture, but if we can highlight on this point for the benefit of the participants. What is the new going on and what can be done in the future?
I am very sorry, sir. I can't say anything on this aspect. Particularly, I am working on this. I am not working on this aspect, millets. I am focusing on the vegetables, particularly, and the remediation.
I am well aware about your topic, but I just wanted as an expert or that what can be your advice to our younger students and in this direction, because that is one of the priority. We try to link SDG and food point of view and the millets, if you have any idea on that. Or anyone in the forum, even I will ask Dr. Richa in the other deliberations, where we can get the benefit from the expert about the SDG points on facet related to food and other aspects. That's my point. So Dr. Richa, whenever you get an opportunity, please try to get the opinion from the experts.
Absolutely, sir. And it's a very important topic to be addressed, especially this year, being the International Year of Millets, as has been said that it can be grown in tough areas, wastelands, and can be beneficial to the general population at a very low cost. And many of the SDGs could correspond to the the advancement and promotion of millets. Certainly, there are many products also that are coming up, thankfully, that are based on millets. And we will see for sure a good future and a good relation with the SDGs being achieved. With such an important, a full complete year has been dedicated to millets, and it's very important for all our, especially botany community and our agricultural community to address the same issue. So we'll take a note of that, and we'll surely ask our experts
thank you.
who can address the issue, particularly addressing it with coordination with the SDGs. Thank you so much, sir. Our next speaker, Dr. Aarti Mishra, is Assistant Professor, Department of Botany, Hansraj College, University of Delhi. Her research area is phycology, environmental microbiology, bioremediation, plant microbe interaction. She has several publications and awards to her credit. Ma'am, we are very happy to have you with us today. We'd like to felicitate you with a virtual Tulsi sapling plant, as is our tradition. So Dr. Karthikeya Shukla would like to offer a virtual Tulsi sapling to Dr. Aarti Mishra. Dr. Aarti Mishra, we welcome you and we're very eager to hear from you.
Thank you, Richa
Ma'am?
ma'am. I hope I'm clearly audible.
Yes, ma'am, you're audible. And it's so good to see you after long.
I'm sharing my screen.
Yes.
I hope it is visible to all.
It's coming up, ma'am. Yes, it's there. But would you like to put it on show mode so that we can confirm for you?
Yes, just give me 1 second. I don't know how to get rid of this panel. Yeah, now. So first of all, I would like to thank the organizer of this organizing committee of this 5 days training program on this management strategies for environmental health safety, a step towards achieving SDG. So it is such an important topic, and I'm going to add little on this by talking on the biodegradation of organophosphate pesticides. Sir was talking about on the metals, heavy metals, and now it is shifted. My talk is completely towards the pesticide degradation, and especially the organophosphate one, by the microbial remediation by using this Actinobacterium Zehengluella species ISTPL4. So which I have isolated in my postdoctoral studies at Jawaharlal Nehru University. So I don't know how to change the slide.
With your arrows, it is not shifting, or with the mouse click? You may try and share your entire screen.
Yes, ma'am, now. And
Ma'am, it's coming up. We'll confirm. It's there, ma'am. Can you try and click it?
You can do it with the arrow.
arrow, it is, I think with the arrow, it's not working. We are trying.
Just click on the
First
screen.
of all, sharing screen, share screen, share,
Click on the screen.
and then move to the next slide. Maybe I think that's a requirement. So is it okay if I do like this? Because I don't know if I'm sharing with full screen, it is not changing actually. Is it visible to all?
Ma'am, it's visible, but it's smaller on the screen. But anyhow, as you're comfortable.
You can try it with the.
Now it is working, I think. Yeah, now it is working.
How do?
Yeah.
How it work?
It's there, it's there.
Yeah, okay. So we start with the, I was talking about the remediation, and now I'm telling about you about the Himalayan region, where actually it has received a particular attention due to their special habitat that is psychrophilic in nature, like ice sheets, tundra soil, glaciers, tundra wetlands, and also different lakes. So what is the environmental significance? So actually the microbes which is present in such conditions, such environment, they have great potential to degrade different pollutants, and also they are very potent agent for the bioremediation. And also they identify as a source of novel bioactive compounds and also cold active enzymes, and they are especially used in the biotechnological and industrial importance. So in such a habitat, this Pangong Lake is one of them. And this lake is actually situated in the border of India and China. So this lake has very high altitude, and it is a brackish water lake, so extreme environment. So why I'm telling all these things, because from where I have isolated this bacteria Zehengluella, so this is the location from which this sample I have isolated. So this lake is situated at very high altitude, and it is very extreme environment over there, low temperature, high alkalinity, and also nutrient level is very, very low. So to identify or to classify the microbes from such an extreme environment, we have used two different approaches. Like one is culture dependent approach, and other one is culture independent studies or independent approach. For culture independent approach, we have done metagenomic analysis. For this, we usually go for the outsourcing, and we just isolate the metagenome genomic DNA from the sample, and then we go for the sequence. We give it for the sequencing, and we analyze the sequence-sum analysis. The company, they already do it for us. And for example, for taxonomic classification, for functional classification, we also compare our sample, our sequence from this DNA with the other known sequences by using different softwares like Kaiju, MGRAST, for the taxonomic classification, and for the functional classification, we use Cognizer and MGRAST. So this is the case of culture independent study. But today I will be talking more about culture dependent study, which I have done, and some of the colleagues from the same lab, they have done. So what we have done, we have screened and isolate and identify different bacterial isolates. So we enrich the culture in different medias, and then after getting different colonies, after doing serial dilution, plating, we got different colonies. And what we have done, we have identified those organisms, especially the bacterial ones, bacterial isolates, and then we have done whole genome sequencing of some of the bacterial isolates. So now coming into detail, as you can see here, so here I have, I was telling you, we got many isolates, bacterial isolates. Some of them we have analyzed, like this P1 is Pseudomonas, P2 is Chivinella, and P3 is Pseudomonas, another species. And as you can see here, the isolate 4 was the Zehengluella species, the bacteria. Then we go for whole genome analysis, and first of all, 16S. Sequencing. After that, we came to know that this isolate is Zehengluella. And actually, this is an actinobacterium. It belongs to the family Micrococaceae of the class Actinobacteria. It is a gram-positive bacteria, small rod-shaped, pale yellow color bacteria. As you can see here, the same picture. The sequence similarity data indicated that this Ziehengluella species has the highest sequence similarity with the other Ziehengluella strain species, halotolerans, and this strain. The 6th species of this genus is already known. So this one is the 7th one. And on the basis of genome analysis, we got to know that DNA G+C content was different from the other known species, that was 69.84, so that we can say that this species, ISTPL4, is a novel species of the genus Ziehengluella. So what we have done, we just go for the whole genome sequencing to know more about it. So we have published this genome in Genome Announcement in 2018. And as you can see here, genome stats, genome size is not much. It is only 3.5 MB. And all the details are here. I'm not going into detail. So far with this bacteria, we have done different bioremediation work, we can say. So first we checked what are the genes present in this bacterium. So we have done with the polycyclic aromatic hydrocarbon degradation, especially the phenanthrene, heavy metal remediation, because this bacteria, they have the metal removal genes also. So heavy metal remediation, particularly chromium. Plant growth promotion activity, his work has done by me in my postdoctoral studies. I have done this work in NIPGR, and also the dye degradation, especially the methyl red dye degradation work I have done by using this Ziehengluella. What is ongoing work? It's synergistic effect of both fungus and actually I'm making a consortia and checking this the plant growth promotion by the synergistic effect. This bacterium plus a fungus, including the fungus. But what more we are doing, that is pesticide degradation by this bacterium. So pesticides, as we all know, that is this pesticides, they are so much dangerous. And it is especially there are different groups of pesticide, organophosphates, organochlorines, carbamates, and there are many these groups. So I have focused on particularly the organophosphorus compounds, and mainly they are mainly the ester or ethyl derivatives of phosphoric acid. And these organophosphorus compounds, they are very toxic to mammalian cells, and also they are having a very adverse effect on the terrestrial, not only on the terrestrial, on the aquatic ecosystem also, but on to human also. They are very actually they are very, very dangerous. In US it is banned, actually some of them, like yeah, so there are many. Actually, this group includes glyphosate, chloropyrifos, parathion, methylparathion. So these are different categories of it, or different types of which are under the organophosphorus compounds. So I focus mainly on the microbial remediation, particularly with this bacteria, because there are some, by whole genome analysis, I got to know that they have some genes, enzymes, like esterase, organophosphate, and these enzymes, they are responsible for the degradation of such compounds. So what I did, I checked the utilization of these pesticides by this bacterium. And for the initial work, I have used chloropyrifos, dimethionate, malathion, and monocotrophs. So these four I have used. And then after checking with the different concentration, we came to know that with different concentration of all of these, we came to know that chloropyrifos, this strain is utilized chloropyrifos most. And among the chloropyrifos, with the different concentration of chloropyrifos, we checked, and the strain showed its growth up to the 900 ppm. So you can see how high it is. And actually at the 600 ppm, the concentration of chloropyrifos strain showed the best growth. Then we optimized by this growth with different pH, different salt concentration, different glucose concentration, different temperature, and different inoculum size. So what we got the result, and then we have used the growth curve stats, and we explain it by two different models, logistic and Gompertz. And as you can see here, at pH 3, salt concentration 1%, glucose 1%, and at the temperature 30 degree and inoculum size 3%, the strain shows best growth. GC analysis we have done to know the metabolites, and there were two key metabolites identified. One is benzene 1,3-bis 1,1-dimethyl ethyl, and second one was phenol 2,4-bis 1,1-dimethyl ethyl. So as you can see here, we proposed a pathway of this chloropyrifos degradation in Ziehengluela, this bacteria. So we thought, okay, we have 4 different putative genes, or putative, we can say putative enzymes for the degradation. And this we got to know when we analyzed the whole genome sequence of this Ziehengluela. So it contained esterase YBFF, carboxyl esterase B, carboxyl esterase NIHH, and phosphotriesterase. So we were thinking which one is more important and which one is actually responsible with this for this degradation. So for that, we used in silico analysis. And before going to the in silico analysis, we wanted to know more about these enzymes. So for that, we used different bioinformatics tools to check or to know more about these enzymes. So what are the number of amino acids, how much the molecular weight. So as you can see here, the molecular weight of this one, carboxyl esterase B is most, it is 55 kD. And also the theoretical isoelectric point, and then it is, so all of them are acidic in nature. So, and also aliphatic index. So it is high aliphatic index that indicates that all the proteins are thermostable. And also the average of hydropathicity, that is also, you can see it is in the minus, but the negative value indicates more surface availability of the protein to interact with the water. So all the proteins are stable. Next is the structural secondary structure prediction. So as you can see here, all the percentage of alpha helix, beta strand, beta turn, and random coil. So in general, you can see the random coil, percentage of random coil is very high in all the proteins. And also the beta sheet, or we can say the extended strand, is less. So active site, we came to know that most of them, I think 3 of them, they have alpha beta hydrolase active site. So to know which one is more interacting with the chloropyrifos, we have performed this docking studies. So in the docking studies, as you can see here, this interaction of these enzymes with the selected pesticide chloropyrifos has been done. And this one is the 3D confirmation of the chloropyrifos. And chloropyrifos molecule, this structure we have obtained from the PubChem website. And Chimera is used for the energy minimization. It's again not moving. Yeah, so we have checked the binding affinity, and all these binding affinity is, I think it is the strength of interaction, how much interaction is there. So we have checked with all these proteins and with the chloropyrifos. And as you can see here, it is in -7.2 binding affinity with the carboxyl esterase and chloropyrifos. So, and that this docking studies we have done with the AutoDock Vienna program. So there are, now I'm ending my slides with the, with telling you the key findings. So strain shows utilization of chloropyrifos, and it shows approximately 78% of degradation. This chloropyrifos is working, degradation is working as optimized. We have optimized it at pH 3, salt concentration 1%, glucose concentration, temperature, and 30 degree, and also 3% inoculum size. GC analysis showed 2 key metabolites, which is written here, and also proposed chloropyrifos degrading pathway has been shown. And also in silico analysis, docking studies, we showed that carboxyl esterase B has the maximum potential for the degradation of chloropyrifos, which is present in this Ziehengluella. So in conclusion, we can say this Actinobacterium Ziehengluella species ISTPL4 is a potential candidate for biodegradation of pesticides, especially the chloropyrifos. So now I would like to acknowledge the work has been, most of the work has been done in Amity, because before joining to the Hansraj College, Delhi University, I was working as an assistant professor in Amity Institute of Microbial Technology. And Himanshi was my scholar, and she has done most of this work. And I thank also Professor Thakur from School of Environmental Sciences, who was my mentor at the postdoctoral studies, and also UGC for giving me opportunity and giving me this Dr. D.S. Kothari Fellowship. And thank you for inviting me and for this talk. Thank you so much.
Thank you, Aarti ma'am. It's always a delight to hear from you and to see you here. And ma'am, what you have shared with us is one of the most informative techniques, and the results are also very, very encouraging. So during the process, what are your observations that what are the means of reducing the pollution or the contamination of chloropyrifos?
Yeah, actually, these first of all, these pesticides should be banned. So, because in USA, if I talk about the chloropyrifos, this one is actually banned in USA. And in India, I think Tamil Nadu is the state first. They have taken an initiative to towards this for banning this, but it is still they are allowed for some of the crops. They are allowed, and yeah. So I think we are also going one more step. Like we are trying to purify this enzyme, and maybe we can develop some of this, I don't know, some technology so that we can use this microbial remediation, and we can actually we can do something for the crops, yeah, to remove this chloropyrifos or
Absolutely.
other pesticides, yeah.
Yes, and I think more and more of your inputs on this research would certainly bring about a workable solution here. Because as much as we know, and we also research in this area, that the pesticide pollution is more than what we see. Sometimes the more of application, sometimes the monitoring is less as to which pesticides are being sprayed and why still some banned pesticides are still being used. So one of
I think
the
10 or,
key,
yes, yeah, I think 10 for 10 crops, as I remember, 10 to 12 crops, they are still not banned in India. So, but it should be banned for all the crops. Yeah.
so true, because once it is there in the soil, it's there. And the persistence can be reduced by such a innovative technology that you have presented today. And we're sure that the cost-effective solutions that you can bring forth for the farmers and agriculture can bring a new revolution. Thank you so much for sharing your research experience and your knowledge with all our participants in this training. Thank you, ma'am, for joining us.
Thank you, Richa ma'am. Thank you.
Now we have with us our next speaker, Dr. Kartikeya Shukla, Assistant Professor AIES, Amity University. His areas of interest are environmental chemistry, animal toxicology, aquatic ecosystem toxicity, and water quality. He has done his PhD from IITR Lucknow with more than 10 book chapters and several publications to his credit. He's currently working in the area of bioaugmentation and remediation of aquatic ecosystem. Sir, we are very happy to have you with us to deliberate on further discussion on SDGs and how they can play a role in each individual's life. We'd like to felicitate you first with our traditional Tulsi sapling. Dr. Kartikeya Shukla, Assistant Professor, Amity Institute of Environmental Sciences, AUUP, is with us right now. And Dr. Anamika Srivastav offers the virtual Tulsi sapling to Dr. Kartikeya Shukla. Hope you will accept the virtual Tulsi sapling, sir.
Thank you very much, Richa ma'am. I'm highly honored to be there for such a huge event where many of my seniors, my colleagues, the senior colleagues, they from IITR have delivered their lectures. Many of them are of such eminent scientists, even are my colleagues. They all have delivered lot many things, see how to achieve SDG. Each one of us is having a different approach. All right. Hopefully my screen is visible.
So your screen is visible, but I think there are certain concerns regarding your voice not clear as the participants are placing. So maybe request you to please see once.
It should be pretty much clear. Is it audible clearly?
Yes, sir, better.
Okay. So we all have talked about different aspects of achieving the sustainable development goals in all the previous sessions by all the speakers. I'm not pinpointing anybody. Everyone was appropriate up to the mark. But what I have focused on is to just raise one question. Do we all understand what actually sustainable development is definitely we are the scientists, we are the academicians, and we'll say it is to manage the resources that we are having today for our future generation. Right? Definitely. And then when we focus on, that is the theoretical part. And when we switch over to the sustainable development goals, 17 of them, if you see, with 169 targets, how to achieve them? Any idea, any what to say, any strategy to achieve all the 17 sustainable development goals with 169 targets? We, the people of academicians and scientists, definitely can understand them and act accordingly. But if I say it is not good enough for the layman people, who are the most appropriate one to work upon. Without their participation, without their intervention, we cannot deal with it. We cannot achieve the goals of sustainable development. Because each one of us, whether it is in one line what I have said already, it is conserving and preparing the resources what we have today for our future generations. That's it. Many of us don't focus beyond that. And for that, focusing beyond our future generation, why not the present generation? And for that, we need to understand what actually sustainable development is. And that's what the topic is. And when we understand, as a common man, not an academician, not a scientist, if I say, so we need to understand what actually sustainable development is. My presentation is not of science. It is something that we need to adhere in our day-to-day life based on scientific research, scientific responses that we have in the recent past, the natural responses that we have. So let us proceed further. And if I say it is the thing, the term that came into existence about sustainable development. So it was in 1980 along with the WCH conservation strategy, which was targeted to achieve a new status by the Brundtland Report on North and South for a program on survival and common crisis, along with our common future of 1983, the report by the United Conference on Environment and Development held in Rio de Janeiro in 1992. Before that, the sustainable development term was not into discussion. If we talk about development, so what do you feel? What actually development is? Though my slides, as for the uh, presentation of Dr. A.V. Pant on the very first day, my slides are overwritten, but definitely it needs to be overwritten here. So that each and every one, because our target participants are our UG and PG students along with our PhD scholars. So they must go through each and every word, because the lesson you forget, but if it is there written in front of you, you try to remember it, learn it fast. So when we talk about development, so generally what we feel, the gradual growth of a situation that becomes more advanced and stronger than previous one, which is intended to bring a positive change for human being and its surroundings. Sometimes it may take place by bringing about a change in policy, projects, and legislation, which in India, at least, we have observed in the last decade. Definitely, amongst us, many of them will not agree that the change in the policy, the projects, the legislation is relevant. But in a longer run, for the purpose of development, if we see something that is significant, that is positive. Definitely. At the very present day, it is not suiting us, but in a longer run for our coming generations, it will be beneficial. Needs to be thought of. Why so? Just because we need to unfold the human potential for meaningful participation in economic, social, political, cultural process and institutions to improve the conditions of each and every individual around the globe. That's what actually development means. And when we talk about sustainable development, whatsoever you do, researches in medical sciences, environmental sciences, biotechnological sciences, nanotechnology, engineering sciences, anything. If it is not having the potential to fulfill the needs of the hour, along with catering our future generations, so it is useless. So sustainable development is fulfill the requirements of the day and manage the things so that each and every bounties of environment of nature that we have enjoyed may be available with our future generations. The first ever concept of sustainable development. We all are environmentalists. I'm just quoting it. Graham Brundtland in 1987 proposed the concept of sustainable development, though it took few years to get it adhered by the global community. He was then the Prime Minister of Norway and Chairman of the World Commission on Environment and Development, where he proposed that there should be a sustainable development, not only the economic or industrial development, it should be a sustainable development where we need to improve our lifestyles and well-being with preservation of natural resources and many of us don't see it. We do it our way. We focus on improvement of lifestyles and our personal well-being. We focus there. Right? In a longer run, if we see, so that will not be acceptable for preserving our natural resources and ecosystems, ultimately affecting us adversely. The evolution of sustainable development was in 1972. If we see, so in 1987, it was discussed, explained at large. But way back in 1972, in the Stockholm Conference, it was discussed initially. छोड़ दिया काय को करना, we need to have economic growth, and that was the time when the developed countries were in full. The report of the World Commission on Environment Development of 1972 is the proof to it. Later in 1992, there was a conference on environment and development for our common future. The theme was, popularly we know it as the Rio Earth Summit, where successive landmarks and events were sought to build the outcome of previous events, and that provided input to the preparatory process of several events. Like we established the Commission on Sustainable Development in 1992 December, so as to ensure that effective follow-up of UNCD, that is United Nations Commission on Sustainable Development, and to monitor and report the implementation of the Earth Summit Agreement at the local, national, regional, international level. Then Rio +5, Kyoto Protocol, all were formulated and implemented, though not accepted by most of the nations, but majority of the nations around the globe, the members of the United Nations, they accepted and they had been working. The major target was, or rather the major aim was to maximize the probability of achieving sustainable development while minimizing the chances of environmental degradation. You see, take into account these 2 points, where come the common population? Because the members of the United Nations are the government bodies, the national representatives. So who come will raise the question, and how come the common person will be involved with this? Until unless the person is informed, well educated, well aware about the aims, the objectives of sustainable development, the benefits and the demerits of sustainable development. And then he needs to be made acquainted with the goals of sustainability. And the best way out is, and when we teach natural resources in our curriculum, so there is a simple thing that we say to our students, and that is the locals are the best placed for the conservation and management of the resources. So why not involve the local public? They may be informed, they may be make aware of each and every policy, not national as well as international. Get them to know what are the benefits they'll be increasing. The politicians, the industrialists may incur their profit. No issues with that. But the local people, the last person who is getting affected by any variation, any change in the environment, in the availability of the natural resources must be informed. And if the very person is convinced he is, the policy matters, the strategy is beneficial, he'll cooperate. Four major components need to be taken care of. Definitely, we all know it, the climate change, the nutrient cycles, the hydrological cycle. The fourth one is very important, the biodiversity. Being environmentalists, toxicologists, naturalists, or biological scientists and academicians, what we feel about biodiversity? All the populations other than man that surrounds man, right? But my dear good friends, man is a social animal and the most devastating one. So the activities and the impact and the effects of man need to be taken into consideration at large, because the climatic change, nutrient cycles, hydrological cycle, the three major ones, the three major components need to be focused, as man is the culprit to disturb them all. Why so? If we see the 4 points here, we get the pillars of sustainable development. Interdependent and mutually reinforcing pillars of sustainable development, which were the outcome of the United Nations World Summit of 2005. We identified three of them: social development, economic development, and environmental protection. Good enough. I hope everyone agrees with it. But for any sustainable development program, policy, legislation, as what I have mentioned here, we need a fourth pillar, and that is cultural diversity, which is necessary for mankind as biodiversity is for nature. Sustainable development cannot be understood and implemented in terms of economic growth, but also as a means to achieve more satisfactory intellectual, emotional, moral, and spiritual existence, as it was discussed in Universal Declaration on Cultural Diversity in 2001. So the cultural diversity makes the fourth pillar along with social, economic, and environment protection. How culture becomes a pillar of sustainable development? When we think of development of a certain area, certain locality, certain geographical region, what we need to understand, what we need to learn is how people think, learn, and solve problems. What are their values, their respect, what attracts them, delights them, and why they get offended. Also, their sense of thinking, what offends them and what is appropriate. You see, the soil in which the tree is having its identity, has its roots, and that needs to be properly observed, properly learned, so that whenever we try to manifest anything, any approach, so that very population may not get affected. The systems of the organization, technology, art, politics, economics, community life does not get adequately affected. Else, the best of the policies and the strategies will fail. And here I have just tried to learn it and sharing it with you all. The metaphor of culture that I have mentioned as the fourth pillar of sustainable development. You see, we have what we do. We have two different components of culture. One is the visible culture and the other one is the primary culture. In the visible culture, it is language, folklore, laws, music, customs, fooding habits. That is the artifacts and the behavior. That may be the traditional one or the acquired one. But actually, in the subconscious mind, in the subconscious heart, we have a primary culture. Wheresoever you go, all across the world, which is highly patterned, implicit rules of behavior exist, and the hidden cultural grammar, the beliefs, the values, the assumptions are still there. So, how to deal with it? That is a very essential point to be taken care of when we are formulating, we are planning any policy or strategy for development in under the umbrella of sustainable development. The primary level culture that needs to be taken care of, this is the prime one, though it is the fourth pillar, but that needs to be taken at the very instance primarily. So that is the underlying hidden level of culture that is highly patterned, a set of unspoken implicit rules of behavior, thought that controls everything that we do. This hidden cultural grammar defines the way in which people view the world, determines their value, and establishes the basic tempo and rhythms of life. Most of us are either totally unaware or else only peripherally aware of this. These are the words of E.T. Hall, which said it is the hidden paradigms of primary cultural level. And in most of the cases, if we see the developmental activities in last, say, 5 decades, we ignored the hidden paradigms of primary culture. And that's the reason Joshi Mutt is declined. We focus on development, but not onto the hidden paradigms of the primary level culture. Before the other metaphors of culture I explain or say, let us see this picture. What do you feel like? Anybody who can mention it in the chat box. Anyone? No, no issue. It seems to be the forehead of a horse, right? Actually, it is not. It is the knee of the foreleg, one of the four legs of a horse. What I mean to explain or what I mean to express by this very picture on the screen is the problems that are visible to us if we don't go into the cultural biodiversity and economical values, economical issues, so such gaps may arise. And that creates a different metaphor for the culture. Means what we need to do for sustainable development towards achieving the sustainable development goals, the collective programming of the mind which distinguishes the members of one group from another needs to be done. That is, the software of mind needs to be analyzed. The eyes through which we see the world. Putting in all together, if we see that is a major focus area for each one of us is to have the economic development. The secondary one is social development. The sizes of the circles or say the ovals that have been made that say all the story. What I want to say, the major focus is on economic development, then social development here, but that should not affect the cultural diversity. And then only we'll be able to protect our environment for a longer duration of time. When we talk about sustainable development, what we think of, few points are there on the screen: clean water and air, fertile soil, good food, a livelihood, healthy economy, optimum population size, halting global warming, safety from poverty and disease, social contact and a sense of community, work, rest and celebration, and opportunity to learn. Ask yourself, how many of these we are having with us? I'm not going to answer you. Answer it to yourself. And then talk about whatsoever the science you do, whatsoever the profession you are engaged in. The 4 pillars of sustainable development. Now, initially they were 3. Now, in the present day scenario, 4 of them are there. Environmental sustainability is the prime one. What? Economic development, social development, and cultural development, which ultimately will result into environmental protection, where we'll have ecosystem integrity, biological diversity, and managed population size. And to achieve the 3 targets, I will say, from top to bottom, we can go. We need to maintain our cultural diversity at the utmost level. Then it may be, it should be social development that will lead to environmental protection that ultimately will give us economic development that will give us environmental protection. Then once it is achieved, so social development, economic development will be there, and finally here, though it will take time, no doubt, but it should be the mindset, the learning and understanding of what actually is required in the present day. This is what we require, and here is what we need to do today. The principles of a sustainable society means at our individual levels, at our social levels, what we need to do is respect and care for the community of life, improve the quality of human life, conserve the earth, vitality and diversity, minimize the depletion of non-renewable resources, keep within the Earth's carrying capacity, which is much beyond. We are requiring another Earth to fulfill our requirements. So that is a bit difficult task. Then changing attitudes and practices. This one uh, is the need of the hour. Enable communities to care for their own environment, provide a national framework on integrating development conservation. We are on to it. And also creating a global ally. Sustainable principles, sustainability principles that we have, it is to reduce dependence upon fossil fuel, underground metals and minerals, reduce dependence upon synthetic chemicals and other unnatural substances, reduce encroachment upon nature, meet human needs fairly and efficiently. Means we need to make a coordination between old and new approaches for human use. And also there has been a commitment by India that by 2050 the net zero carbon emission will be eliminated. Hopefully. But how come? That is a commitment of government of India. If not, we are into it. We as an individual, not an academician, not as a scientist. Let us see to what level we achieve it. And for that, whatever the experts say, I'm not the expert of sustainability, sustainable development. I'm fond of learning, reading, and that's why I chose that very topic. So sustainability issues need to be analyzed at various levels. From bottom to top, I'll go through it. It is the soil losses in terms of physical, chemical quality of the soil. Then at the local level, to what we do around us, air, water, soil pollution, or waste generation, anything. Then the regional, national, and area level where water pollution, water depletion, deforestation, fisheries depletion, biodiversity, desertification, erosion, everything is considered. And then the global level key where we discuss about ozone depletion, global warming, climate change, air pollution, and lot many things. So what we need to do, what we need to act upon here. Hopefully, I'm pretty much clear and understood by each one of us. Our goal of sustainable development is the active involvement of the government, NGOs, private sectors, along with civil society. Not just politicians or policy makers. That should be. We need to remind policymakers to make decisions to protect our future. Remind them that we want better jobs, cleaner cities, more equitable resources distribution, and above all, a secure future full of opportunities, a pleasant and healthy planet to live in, not only for us, but for our future generations. But the major problem is the poverty and population pressure. Developed nations, they have the economic resources where we can procure the natural resources to fulfill their requirements. And they are less populated. But what about? the developing nations, economically weaker nations. So this intersect between poverty and population pressure each time humans and their environment suffer. In general, or to sum up, if I say, this will be the last slide, though I have taken much time. I think it is 10 minutes over. So to achieve sustainable development, what we require on individual level, maybe the scientist, the academician, a layman, whatsoever, our activities may be the eco-friendly one. Each one of us should be aware of the current needs, that of the future generations, ensure productive asset availability to future generations. Technological development may be in a way that no component of the environment is harmed and help restore the natural environment wherever feasible. That has been summed up in the picture next to the text. And for all these, though, in last 2-3 decades, 2.5 decades, we have a lot of many green technologies, so-called green technologies that are not disrupting, that are not disturbing the environmental components and everything. And definitely they are. So they need to be disrupted. Even better options are to be explored so that our future generations maybe at least they may enjoy the bounty that we have enjoyed in our childhood. Thank you a lot for the patience.
Thank you, Kartikeya sir. It was a very unique and a very different approach to addressing the issue of SDG and how it has to be known to everyone, and one has to be sensitized to more towards which kind of approach one is having for the SDG solution. So thank you, sir. It has been one of the most informative lectures for all our participants, I'm sure. Thank you so much, sir. And we would like to thank all our speakers for the day. Dr. Elora ma'am has so wonderfully addressed the issue of cadmium toxicity and how it is a system toxic substance. It can lead to so many different problems. What are the different mechanisms, physiological mechanisms that come up with the cadmium toxicity? We had Dr. Rajesh here with us from BHU. He also addressed the heavy metal pollution, the different aspects of different indices related to heavy metal pollution, and how we can have bioremediation as a very important technique to tackle the issue of heavy metal contamination in our environment. We had Dr. Aarti Mishra from JNU. She also expressed her views, and her deliberation was on the research areas centering around chlorpyrifos and its bioremediation techniques with a microbe that has been wonderfully innovated out. Dr. Kartikeya Shukla then addressed the participants over the issue of sustainable development goals, what approach should we have for sustainability, and how true should we be to bring out the solutions and be honest to our sustainable development goals. We are very thankful to all our speakers for the session 6 today. And we're very thankful to all our dear participants for patiently listening and putting up their questions to all the speakers. It has been a wonderful session to deliberate, to discuss, and to be enlightened with so much of experience knowledge today. Thank you to all. We would meet again at 2:30 sharp for the next session, the section 7. Thank you all, and we look forward for you to joining us again for the 7th session.
ଯାହା ପାଉଛି ତେଣେ ବଦମୁ
Welcome to the 5 days training program on management strategies for environmental health and safety, a step towards achieving SDGs organized by Amity Institute of Environmental Sciences in association with the Indian Institute of Toxicological Research, Lucknow. I am Charu Singh, the MC for this plenary session 7. On behalf of the organizing committee, we extend our appreciation for your presence today at this deliberation. The theme for this afternoon session is Pollution Prevention and Sustainable Solutions. Ladies and gentlemen, we feel honored to have with us our esteemed speakers for the session. I welcome Dr. Pratibha Gautam, Dr. Manoj Chandra Garg, Dr. Vinay Kumar Khanna, and Dr. R.D. Tripathi. My heartfelt gratitude to all the dignitaries, resource persons, and participants for joining us back in this session. In today's session, we will talk about pollution prevention and sustainable solutions. Technological progress improves all aspects for our lives, but it comes at a high environmental cost. The manufacture of consumer goods, the production of energy, and even the development and utilization of effective medicines pollute our water, soil, and air. Society has also developed powerful pollution remediation technologies. However, these tend to be very costly, but in economic and energy terms, and often result in pollution of different forms. This pollution cycle is clearly unsustainable, and it has become essential to develop integrated approaches to control pollution that do not add to the problem. In today's session, we will be discussing about some of the most relevant aspects in terms of pollution prevention and providing sustainable solutions for the same. Participants can put up the questions during the presentation and type them in the chat box. We will have separate time for question answer session after every presentation. Now let us move forward and begin this informative session. It's my pleasure to introduce the first speaker for the session, Dr. Pratibha Gautam. At present, she is working as a consumer safety associate with the Global Safety Assurance team at Reckitt Benckiser. She received her doctoral degree from DRDE Gwalior, India in 2012. She has worked as a project scientist at IIT Kanpur, a Department of Biological Sciences and Bioengineering. Dr. Gautam is a reviewer of research articles published in Defense Science Journal. She has also received travel grant from INSA and MPCST for attending the conference at the International College of Integrative Medicine, Pittsburgh, USA. She has been a member of Society of Toxicology India, Society for Neurochemistry, and Society for Mitochondrial Research and Medicine India. Ma'am, I now request you to please address the gathering on the topic pollution prevention and cleaner production. But before that, we would like to humbly facilitate you with virtual Tulsi sapling. Dr. Anamika Srivastava is doing the honors. Welcome, ma'am. Over to you.
I think ma'am is facing some technical glitch because we cannot hear her.
Yes, ma'am.
But she's here. I think she's facing some net issue, Dr. Amrina.
Yeah.
Pratibha ma'am.
Uh,
Pratibha ma'am, can you hear us? Ma'am, your mic is on, but we cannot hear you.
Pratibha ma'am.
Pratibha ma'am.
Pratibha ma'am.
Yeah, hi. Sorry for the inconvenience. There's a technical glitch here.
It's okay. No issue, ma'am.
Yeah.
Welcome to Amity University online platform, ma'am. Welcome. Namaskar.
Thank you so much. Namaskar. Let me start with the presentation then.
Okay, ma'am.
Yeah, I'll just open it and then share the screen. Would you mind if I switch off my camera and then start it? Because maybe there's a technical glitch.
Okay, ma'am. No problem, ma'am.
Okay. So, hello everyone. I did not attend my introduction, so I would like to give my introduction to everyone once.
Pratibha ma'am.
Yeah.
Ma'am, just give me a few minutes. We will introduce you. Just give me a few minutes, ma'am.
No issues, no.
But it's our pleasure to introduce, ma'am. Just give me a few minutes, ma'am. Only 1 minute, ma'am. Ma'am, please unscreen your PPT, ma'am. Unshare your PPT. Ma'am, please unshare your screen, ma'am.
Yeah, sure. Just
Charu.
Yes, ma'am.
Charu, please again start from the starting. Start the session.
Yes, ma'am.
And introduce ma'am.
Okay, ma'am.
Sorry, I'm unable to stop.
No, ma'am. It's fine.
Yeah, now please go ahead.
In today's session, we will be discussing about some of the most relevant aspects in terms of pollution prevention and providing sustainable solutions for the same. Participants can put up the questions in the chat box. Now, it's my pleasure to introduce the first speaker for the session, Dr. Pratibha Gautam. At present, she is working as a consumer safety associate with the Global Safety Assurance team at Reckitt Benckiser. She received her doctoral degree from DRDE Gwalior, India in 2012. She has worked as a project scientist at IIT Kanpur at Department of Biological Sciences and Bioengineering. Dr. Gautam is a reviewer of research articles published in Defense Science Journal. She has also received travel grant from INSA and MPCST for attending the conference at the International College of Integrative Medicine, Pittsburgh, USA. She has been a member of Society of Toxicology India, Society of Neurochemistry, and Society for Mitochondrial Research and Medicine, India. Ma'am, I now request you to please address the gathering on the topic pollution prevention and cleaner production. But before that, we would like to humbly facilitate you with virtual Tulsi sapling. Dr. Anamika Srivastava is doing the honors.
Thank you so much.
Welcome, ma'am. Over to you.
Okay, thank you so much. I will switch off my camera for a while presenting.
Okay, ma'am.
And then after, when I complete, okay, everybody can see in my screen?
Yes, ma'am.
Okay. So as Charu told us that I am working at Reckitt Benckiser. So I'll be giving a industrial perspective of pollution prevention and cleaner production today, because I'm working with an industry right now in a corporate setup. So continuing with my presentation. So for the objectives of my presentation would be to talk about the pollution prevention, then what are the different responses to pollution, how principles of cleaner production and its benefits help the environment as well as the consumers, then implementation of cleaner production by industries, methodologies that are used for cleaner production assessment, then activities of cleaner production centers. So talking about pollution prevention. So talking about the objectives first, at the end of this session, we will understand what cleaner production is and what sets it apart from other environmental management tools, and how cleaner production can be used as hands-on tool for environment improvement in the industrial setup. We will also understand the challenges and enabling some measures that are associated with the cleaner production, and what are the motivational factors for industry to make use of cleaner production. So now talking about the pollution prevention, basically pollution prevention means reduction of the pollutants or effluents from the source. So it is also called as source reduction. So reducing the amount of any hazardous or toxic substance that enters into the waste stream, or it is released into the environment, including the fugitive emissions, prior to recycling, treatment, or disposal, is called pollution prevention. Then it helps in reducing the hazards to public health and the environment associated with the release of contaminants. The other practices that include the pollution prevention are increased efficiency in the use of raw materials and energy, water, or other resources. Or we can also use natural resources, and we can also do it by protecting the natural resources by conservation. Now, how pollution prevention is done by industries? Pollution prevention in industries is basically done by updating themselves, by modifying the equipment and technology that they are using for the production of a product or a number of products, whatever they are manufacturing. So the process or procedure modifications, or for example, reformulating a product or redesigning a product. For example, during a study, I'll tell you one example from my experience. I will not tell the, due to legal reasons, I will not tell you the name of the product and everything, but I'll tell you the example with a product A. product was being reformulated because there was a component in the fragrance of that product that leads to skin sensitization or skin allergen. So to remove that particular ingredient from the product, it is not only one product that is being affected with that allergen, but there are many products that are being associated with it. So the industries are very particular about each, specifically the company in which I'm working, they are very particular about the pollutants, the skin sensitizers, the allergens, or the or any harmful effect of the chemicals that can cause any deleterious effect to the consumers. So they're very consumer centric. So next one is substitution of raw materials. Substitution of raw materials, that means the fragrance that was being used and had that allergen has been replaced with some other raw material so that we can get rid of that particular skin sensitizer. Then maintenance, training, or inventory control. After that, it is a big task to maintain that, and then we have to train people. It is not only one person or R&D who is responsible for it, but we have to train people and maintain an inventory that what fragrance was being used and which has never to be used in the future again. Now, to consider in-process cycling, a pollution prevention activity must serve a productive function with the making of commercial product for which the original process was designed and must be an integral part of that process. In that, the example that I'm giving, in that we are not changing the whole process of that product. We are just eliminating a part of it, right? So the production process cannot function without the recycling process. That means we have to take care of the contaminants also that we can recycle or reuse in any other product that is not used for the consumer, for that is not used by the consumer by their hands or by that is not going to touch their skin and causing skin sensitization. So in that case, we will not just remove that allergen or that fragrance, but we can use it in some other formulation wherein that formulation is not used by the consumer by their hands or by any of their body parts. Now, the pollution prevention does not include energy recovery, treatment of the waste stream, because the pollutant has already gone to the waste stream, so that is not prevention. Then disposal, any practice that alters the hazardous substance, pollutant, or contaminant once it is generated, because it is already being generated, so that is not a part of prevention. Then practice that is not necessary for production, practices that create new risks to human health or the environment. Now, there are various responses of the businesses to pollution. There are very small scale businesses which do not ever react to the pollution they are creating, but there are very sensitive industries, and they take care of the pollution or emission that is taking out of their industry. So they take care of it. So it depends from businesses to businesses what kind of business they are running and how sensitive they are to their business and the pollutants that are being generated by their company. So first, we come to the passive one, where the business is ignoring the problem, which may usually lead to disasters sooner or later. Now, second, we come to the reactive one, diluting of the pollutant concentrations by discharging to streams, rivers, and oceans. They dilute their effluents into the streams, rivers, and oceans, which can also lead to disasters, but that happens at a later stage and at a magnificent scale later. For example, if you have heard about the Black Triangle in Europe, that is a massive study In Central Europe, there are areas that cover large parts of Poland, Czech, Slovakia, Germany, and Hungary, where long-term air pollution from the coal power plants lowered the pH value in water and soil to a level that now many plant species cannot survive there. Next, coming to the treating the pollution by end-of-pipe technology after the wastes have been generated. This is usually effective, but it is an expensive one. Then preventing pollution waste generation at the source itself, that is through preventive approaches such as cleaner production that I'm going to talk about in my latest slides. This cannot entirely replace the end-of-pipe solution, but in most cases, it is able to drastically reduce the volume of waste pollution that need treatment and also the associated costs with it. At present in our country, on the prevailing approach to how to address environmental problems, at company level and at the government level, there are many steps that are being taken. Now talking about the cleaner production. This is a standard definition given by United Nations Environment Program, that cleaner production is the continuous application of an integrated, preventive environmental strategy towards processes, products, and services in order to increase overall efficiency and reduce the damage risk to humans and also the environment. This is summarized in this flowchart. So the cleaner production means that it is not a one-time project. It is a continuous environmental management approach. It is not only that once the cleaner production is being done and then we can just lay it off. We have to go on, we keep on doing environmental management programs so that it keeps on going continuously. Then coming to the preventive one, it seeks to prevent pollution from being generated rather than cleaning it up after it is being generated. The simple reason is that it saves costs, because if we prevent it, the effluents are not being formed and therefore it saves the costs. Coming up to the integrated one, to be effective, cleaner production should be integrated into the daily operations of the company. In the same manner, just as companies do not do economic management just one time, economic management goes on and on. Every time there are so many meetings for economic management, but similarly, this has to be ongoing, that means continuous. Processes, products, and services, these are opportunities to improve the environmental and social economic performance of the company, which can be identified through the cleaner production. Only by adjusting how things are done, that means the processes, but also what is done to form the products and what systems, that means what services are being applied. The end result of this cleaner production not only benefits the environment, but also the safety and health of the consumers and also the staff of that particular company. Now, although it is not included in the definition, but cleaner production also generates significant economic savings for the company. Now, it is the name that UN and many organizations are using right now, but in some parts of the world, it is known as eco-efficiency, waste minimizations, green productivity, and pollution prevention. These are by and large different names with the same approach. That means cleaner production is the same approach as all these waste minimization, pollution prevention, and green productivity. For a moment, if we forget the formal definition of cleaner production and just think about what it really is, cleaner production, it is a practical tool to improve the production efficiency. For the environment, for the environmentalists, this is same as reducing the environmental impact. For a company personnel like me, this is the way to improve the economics of the company by carrying out these cleaner production assessment in a company. We generate cleaner production options by improving the efficiency. By implementing these cleaner production opportunities, the company will generate less waste. And after generating the less waste, we make better use of the resources that are managed in the process, and which leads to better productivity. This slide summarizes some of the additional features of cleaner production. Let me explain about the relation between cleaner production and environment management systems. Cleaner production is a practical tool, which it is a practical tool to achieve efficiency improvements both in the economic and the environmental setup in a company. It, however, does not define where the company is now or where it will go. It doesn't determine any goals in terms of environmental performance. It does not need any external certification or approval, and can be implemented without any additional cost. But EMS, that is Environment Management System, is a navigational instrument for the company. It defines where the company is, how is it performing environmentally in relation to legislation and stakeholder expectations, what the environmental policy is of the company, and where company wishes to go in the future, for example, to achieve certain environmental standards or goals. Environmental management systems can also be certified through ISO standards. I hope everyone knows the ISO standards, which requires external certification and is often very time consuming and costly. Cleaner production can be an important tool to set up the goals in an EMS, and also it is a first step for a company to get an understanding how it is performing environmentally, if at all that company is not is an ignorant one, that the one I told you in the previous slide, where they ignore pollution. If they are just starting with cleaner production, then the smaller companies can use cleaner production and later develop into the environment management system, because cleaner production is comparatively a simpler tool that does not provide all the advantages of an EMS, but nonetheless it generates substantial advantages of the company, both from if we talk of the environmental and the economic perspective. So in that case, it is always a win-win factor for a company. Then economics of cleaner production. A very important feature of cleaner production that needs to be highlighted is that there is a common misunderstanding that environmental care always comes with a cost. It may be true in case of end of pipe solutions, for example, in case of treatment of the waste, which usually require investments and will not generate any financial payback to the company. While if we talk of the cleaner production, normally generates savings for the company, which means that cleaner production investments usually have a payback. Often the payback time for this investment is very short, but as mentioned in previous slides, cleaner production cannot always eliminate pollution and waste generation, but at least it minimizes it. Therefore, end of pipe solution should not be discarded entirely. And we should realize that there is a hierarchy to pollution reduction. First, there is prevention, then treatment. And in that way, the treatment costs are also reduced. For if we prevent it, there are less effluents in the system. And then the treatment costs are reduced as the amount of waste requires treatment that has already been minimized. So cleaner production has many advantages. But for industrial setup, what are the advantages? That it improves products and services. It lowers the risk of getting caught by legal authorities for environment, then improves the company image. It improves the workers' health and safety conditions, reduces waste treatment and disposal costs. It can be integrated with business EMS. And then it saves costs on raw materials, also energy and water, because we are changing the raw material in case where there are any deleterious effects of those raw materials or the impurities present in those raw materials. And most important, that is for the companies, it makes it more profitable and competitive, because the companies are always thinks of their profits and their competitiveness with their peers. Now now implementing the cleaner production. In this and the subsequent slides, I will be explaining in what areas cleaner production options and measures can normally be identified, and how these can contribute to the improved production efficiency and the reduced environment impact. These options can be divided into 5 categories. First is input substitution, that is changing of the raw material from toxic to more suitable material. Second one is the technology change, that is the better process control and adopting new technologies. Then good operation practices. Good operation practices include improved worker skills, good production management. Then there is product modification, a little modification, change of likewise example that I gave, that changing of the fragrance. It may modify a product a little bit with respect to the fragrance. Then on-site recovery and reuse. All the losses in the productivity, risks and inefficiency can be categorized in all these areas that I'll be talking of in the subsequent slides. This is the basic slide. Now the changes will go on with all the points that I've told, all the 5 categories that I've told you. First coming to the input material substitution. And this consists of options relating to change in the input materials to materials that are less toxic or are renewable or have a longer service lifetime in production. Many companies they have the habit of using low quality raw materials that they purchase at lower price, but it contains impurities that may lead to pollution and some harmful effects to the consumers. But a cleaner production option helps to pay more for better quality materials that generates better yield, fewer production disturbances, and sometimes the product is also of better quality, because if there are less impurities in the raw material, then it can lead to a better product. Then use material for this, all I have covered. Then if we take it to the production process, the products are more, a fine product will be obtained, which will improve the image of the company that I was talking about. Now, the second category is the technology change. It can be divided into new technology. This is often refers this new technology. This is often referred to as new cleaner technology. Cleaner technology is a part of cleaner production, but there are many more improvement options in the cleaner production than only the technology change. Only the technology change will not aid into the cleaner production, but there are other factors as well. The first one that I told you about, the change in the input materials. Then equipment modification, modify the existing production equipment and utilities in order to run the processes at higher efficiency and at lower waste. And emission generation rates. Then improved processing conditions, modify operational process procedures like pH, flow, pressure, dosing. All these are modifiable operational procedures. Then equipment instructions, like for how long the if you know that while doing any experiment, there is a particular time for any reaction to take place. Similarly, there is a particular time for the formation of a product, and that particular time should be maintained. So that should be done using the equipment instructions and or process record, keeping in order to run the processes more efficiently and at a lower waste, so that they generate less waste and emission generation rates. Then replacing, then and improve, which leads to improved process control and improved equipment layout. Coming up to the third one, that is good operation practices. The improved equipment processes. This is the third category that involves cleaner production, that refers to managerial and operational actions which may prevent the leaks and spills and to enforce the existing operational instructions. This is an important category. This is a very important category because of its strong effects, usually without any need for major investment. Human behavior is, because there are workers there in the factories, those who are working, so that their human behavior is the main reason for inefficient production. So good operational practices start with efficient production. For example, in a dairy plant with multi-purpose production and bottling equipment, first producing the straight and colorless products, regular milk, and then the sweet, sour, and color products like custards, yogurts, fruit desserts, or chocolates. So to reduce that, the equipment cleaning needs to, in order to prevent cross-contamination of the incompatible products. So here lies the human behavior. If those workers are not trained enough, that they are not cleaning the instrument or the equipments properly, then this may lead to contamination, and that contamination leads to lower grade of the product. So and eventually it creates and therefore they need a separate production management. Especially avoiding high peak energy consumption can save money in terms of tariffs and in terms of avoiding the need to install high load equipment. Preventing unnecessary power consumption by switching off equipment when not in use. There is a well-planned maintenance program in every industry for all equipment and facilities, and this is very important in this category because without the proper equipment and instruments, industry cannot work. Then rundown equipment is normally not able to operate at full efficiency. So therefore, regular review and maintenance of the systems is a very good example that can save substantial energy and also the costs in many operations. Proper maintenance and cleaning of the equipment has to be the day-to-day routine that results in a clean and tidy space, and therefore a hygienic product if there is a case of a food product. And also hygiene has to be maintained in any setup as far as it is concerned regarding me. Then coming up to the next one, that is the product modification. This is the fourth category of cleaner production option, and it leads to the modification of the product characteristics in order to minimize the this refers to the managerial and operational actions that prevents the leaks and spills, and I'm sorry, this is the product modification, right? That pertains to the modification of product characteristics in order to minimize the environmental impacts of the products during or after its use and to minimize the environmental impacts of this production. For example, the paper products with lower degrees of whiteness, if not required by the customer, minimize packaging, or the mobile phone, or for example, the mobile phones that are designed to allow easier disassembly and recycling of different components. So this can lead to reduction of the harmful substances. Then coming to the reuse and recycling. This is the fifth part of the cleaner production option and is found in the reuse of the waste either in the same process. This is done in the same process or for another useful application within the company. So like for example, I told you that the allergen, the fragrance that was having allergen is not used in the cosmetic products, but can be used in the products that are other than cosmetics and are not going to harm the human beings. So the opportunities for the recycling of wastewater, surplus energy, and chemicals can often be identified with this. This category also belongs to the transformation of waste products into the useful products. Useful products those useful products may be the byproducts of the system. Here it is considered as waste in one company. That means the waste in one of the system and one of the product can be used as a raw material for another product. Then coming to the next one, now coming to the barriers of cleaner production implementation, because it is not very easy to implement. It is easier said than done. So there are many barriers to cleaner production implementation. despite the fact that cleaner production often is called a win-win approach, but it is we should also know about the problems and barriers that hamper proper acceptance and implementation of the cleaner production. The barriers that can hamper the cleaner production process, the internal barriers I'm talking about, the business philosophy or environmental attitude of the CEOs, because CEOs are basically, they are not scientifically sound, but their management skills are very sound. So they may not always know all the pros and cons of the ingredients or the harmful effects of the ingredients that are being used or the byproducts that are being formed in the company. So first one is the traditional philosophy of the environmental attitude of the CEOs, that environment will only cost money, and there's a lack of finance, manpower and time, and especially and their thinking is that it needs special experts that have knowledge, and they are needed to organize and implement a cleaner production options in the company. And the only technical innovation, there are only technical innovations that can reduce waste and emissions. So due to all these initial constraints, internal organization and communication, gaining management support, setting up a project, then limited authority for cleaner production, indifference of the workers. There are many reasons why the CEOs of the company do not agree to the environmental programs or cleaner production. There is very limited information, data and expertise on waste and emissions, and they do not, because they want to be a profitable organization, that is why they do not hire the people that are expert on waste and emissions. They think that resource will cost more to them, the expertise resource will cost more to them, so they do not hire those people. They do not focus on the end of pipe solution, and they think of only the short-term profits that they receive. Short-term profits, that is the monetary profit that they are receiving. Then there are inadequate profit calculations by the cleaner production options. Then missing out this is very important, missing outdated or reliable process instrumentation. And not only the instrumentation, but also I would say the documentation of the chemicals that are being used. In case of Reckitt that I'm talking about, there's a very legal thing that we should see, that the documents that are being provided should not be outdated or unreliable, because in case of chemicals, there's always an expiry. So outdated chemicals should not be used. Then there is no environmental management system to achieve the continuous improvement. They may use it for once, but they do not continue it for long. Then the external barriers that leads to that may not lead to implementation are availability of investment capital, because the small scale industries, they have less capital and they do not try to acquire the cleaner production technologies. Now talking about the cleaner production methodology. So to avoid or to overcome barriers and to guarantee a successful implementation, there should be an organized approach. The cleaner production assessment can be, we can say that it includes 4 basic steps: planning and organization, that obtain further management commitment, identify assessment focus area. Often when there is a limited assessment focus, that is useful to apply for each assessment. Example, there's a section within a factory or a raw material group energy or water. Then to organize the project team, then identify all the barriers and solutions, and then set objectives, and then pre-assess the whole setup of the cleaner production. Then we should understand the then we should identify the sources where it is required, to analyze the causes why it is required, and generally how. This is very important, how to generate possible options to obtain a cleaner production. Then if we talk of the feasibility and analysis, we should check the options of technical, environment, and economic feasibility, and then select the best option. Then how to implement it, how to implement the cleaner production. First, the option of implementation, then option of implementation, that is if at all we are being allowed to do that implementation or not, then only we can move forward. Then we monitor and evaluate the implementation of the project. Then if that project works and is going forward proper for a cleaner production, and there are less emissions, less byproducts that are harmful, then we should sustain it and continue, which leads to successful implemented cleaner production projects. Now, this is a continuous CP loop. Likewise, that I was saying, that it is not only once you have achieved a cleaner production and you should stop, and then you should stop it, and then you can move forward with your production, because once you have achieved that cleaner production, because every time the raw material supply, there are many raw material suppliers that keeps on changing, and every raw material supplier has different impurities, different composition of the raw material. So it has to be a continuous process. When you start a cleaner production project, first there should be a project organization, then assessment. In the previous slide, like I was talking about, then there should be assessment. Then we should talk about the cleaner production options, how feasible it is to use those cleaner production options. Then there has to be an assessment report, whether that is going to lead to cleaner production, or there has to be any amendment into it. Then we implement the projects, we measure the progress, get a final report out of it, and then that final report is submitted to the top management, where it is reviewed, and we check whether the clean production project, how it went, and whether it is leading to the cleaner production or not. So likewise, and there can be refinements that can be made after these reviews, and this all leads to cleaner production. So these are few of the centers, the global cleaner production network. The blue ones are the national cleaner production centers, while others are the CPC and network members. Then these are some of the sectors that are concerned with the cleaner production, like automobile manufacturing, bakeries, breweries, all of these. And the services provided, these are the services provided by some of the cleaner production agencies, that environmental reporting, impact assessment, all these. Not going into the detail of this, but yes, all these are service providers. Then the conclusion. This CP, talking about the CP trends, cleaner production is not only spreading geographically, as you have seen in that map that I have shown, and into new areas of application, but these cleaner production are also evolving into new sectors and levels. Likewise, in the previous slide that I have shown, there are various service providers for this cleaner production. This slide presents some of the current trends in the cleaner production involvement in the developing countries like ours. In many countries, cleaner production was originally introduced through projects in specific sectors, with the capacity building taking place in a few centers of excellence at national level. But over the time, government is also adjusting its policies and regulation to support a cleaner production. As an alternative or maybe a complementary approach to the end of pipe approach, because at end of the pipe approach, it has already gone into the waste system. So that is it is better to use a cleaner production. So cleaner production is also becoming linked to other other environmental management tools, such as just as I talked about, environment management system, and so that we can achieve mutual strengthening. It has traditionally been seen and applied as a tool for improving efficiency in manufacturing industries. Then and the concept is also making making roads to the other sectors, such as municipal services, transport, and healthcare. Therefore, if we say, in order to achieve efficiency improvements in larger industrial clusters, not only in the individual company, which is a traditional focus of cleaner production, because it is said that it is generally seen that the companies that are the small scale industries are mostly involved in this environmental pollution. But cleaner production is also promoted as a tool for environmental management of industrial estates and similar industry clusters. Therefore, for the wider diffusion of cleaner production, it is not only taking place through large centralized coordinated efforts, but more and more it is being promoted at the local level through local industries, associations, and also the local authorities and local stakeholders. Thank you so much for listening to me patiently. Thank you.
Thank you, ma'am, for such a descriptive and enlightening session. Ma'am, we have few questions from participants. So, ma'am, with your permission, we can take up the questions, please.
Yes, please. Yes, please. Go ahead.
Ma'am, first question is, do you think cleaner production as a concept is accepted in Indian industries or is still in budding stage?
We should say that it is in budding stage, because every industry does not accept it initially, but when they are being told, because there has to be some forceful implementation of this, because there is no forceful implementation of cleaner production. So it is still in the budding stage, I would say.
Okay, ma'am. Thank you, ma'am, for the taking out time for your busy schedule for our participants. It was truly a privilege to hear from you. Thank you, ma'am.
Thank you so much. It was pleasure being at MIT and giving a lecture at MIT. I have run out of time, I think. So it was so long. I'm so sorry for that.
No, ma'am, it's fine.
It's okay. Thank you so much.
Thank you, ma'am. Moving ahead, the next dignitary to honor us with his presence is Dr. Manoj Chandra Garg. Dr. Garg is an assistant professor in Amity Institute of Environmental Sciences, Amity University, Noida. He received his doctoral degree from IIT Roorkee in 2015. His key interest areas include water quality, groundwater treatment, membrane filtration, solar desalination, water and wastewater treatment. I now request you to please address the gathering, but before that, as per Amity tradition, we would like to humbly felicitate you with virtual Tulsi sapling. Dr. Juhi Gupta is doing the honors. Welcome, sir. Over to you.
Thank you, Charu. Am I audible?
Yes, sir. You are audible.
Okay, just a minute. I'm just sharing my screen now.
Okay, sir.
Is my screen visible?
Yes, sir. It is visible.
Okay. Thank you, Charu. And I just want to thank the organizer to give me this opportunity to speak on the 5-day training program. So in the last last hands-on training program, I have discussed about some tools, environmental tools, ICT tools that is basically helpful in the field of environmental sciences. I discussed about the like a windrows diagram, eDraw software that basically used for the for drawing of the for flow diagram, for the process diagram to draw the process diagram. And I discussed about some Android-based app like Soundmeter and Luxmeter that you can easily download it from the your Google Play Store. And I also talked about the Excel sheet related software for the modeling, population modeling in the environment, for the Piper diagram, for the drawing of the Piper diagrams and different type of diagrams. And I also discussed about the design expert software that is basically used for the environmental process parameter optimization, multiple parameter optimizations, and origin software that is used for the generally used for the researchers and the students for the plots and EndNote software that is very good for the referencing material, for referencing software. And apart from the EndNote, we can also use the Mendeley and the Jotaro software also. Monavi is a very good software and it's really available for the video editing, and Office Form is used for the assessment. Apart from these, today we just discussed some few more softwares and tools that basically used in the environmental sciences. Okay. So basically, nowadays the artificial intelligence is a very good topic and platform on which we can optimize. We can basically optimize our process parameter, and we can use this machine learning and artificial intelligence in the environmental sciences. Okay, so basically that is a data analytical methodology that teaches machine to learn from the experience, and the natural pattern of the data is discovered by the machine learning algorithms that provides the understanding to make the predictions, and sets of statistical procedures are used in the data science. So basically, in the machine learning, the artificial neural network is basically used. And in this model, we generally used a black box, okay, in which we just give the few inputs and give some outputs, and just and align some neurons in the machine learning in the artificial neural network. And based on it, we train our this network. And based on the training, it gives some models. It gives some models, and these models is basically trained model that predict your validate and predict your testing data. Okay, and based on this, we can predict some water quality and the air quality data in the field of environmental sciences. It is also implemented. Okay, so it's a collections of the connected unit of the nodes is called the artificial neurons, and which loosely models the neurons in the biological brains. So there are different type of neural networks are using nowadays. ANN is also a very old techniques that platform you can use the MATLAB and the other softwares also. Now the CNN and the RNN can also be used, convolutional neural network and the recurrent neural networks, for the predictions of the figures and diagrams also. Okay, so we used these machine learning tools for the forecasting of the air quality and the water quality data. All right, and successfully implement these machine learning tools in the predictions of the air quality and the water quality data. For example, This is one example in which you can see there is some few input data in the green colors. Okay, and that can be the PM 2.5, PM 10, ozone, nitrogen oxides, sulfur oxides, and carbon monoxides. So these are the sets of data that we have getting from the some freely available online platform sites. Okay, and based on these data, we have calculated the overall AQI of the data of the different dates. Okay, and 70% of data we just feed as the input data, and 30% of data we used for the validation purposes, and some few data are used for testing purposes. So based on these, we got a good results in the form of predictions of the air quality. The point R value is become 0.99. That will tells you what the significance of the model. Okay, and so we use these techniques in the predictions. So overall AQI using the year 2020, 2019 and 2018 models. All right, so another performance was discovered when the ANN values obtained via the seasonal models from 2018 to 2020 as compared. And you can see there is a different trend we obtained from these data in the form of winter, summer, springs, monsoon, and autumn seasons. Okay, so this is the one tools that basically used in the environmental sciences, artificial intelligence, or you can say machine learning or ANN, whatever you want to say. Okay. After that, I just discussed about some virtual labs that we are using in the environmental sciences. That is basically one vlabs.co.in is a website. I just pasted a link of this website on the chat box. Just a minute. Okay, so this link is there in chat box. You can open this link and you can see there is a virtual lab developed by the Ministry of Education under the National Mission of the Education through the ICT, Ministry of Education GOI. And that is the link is the vlab.co.in. And the different is having a virtual lab of different subjects. In case of our, we have environmental engineering labs that is developed by the NIT Surathkal. In this lab, we have different lists of experiments, and we also have the theory of this each and every experiment and some pre-test and the post-test also is available in the form of MCQs, and procedure is also given, and the simulation of each and every experiment is also given in this virtual lab. Okay, so today we will do a few experiment. Okay, you can open and you can go along with me in the hands-on training. All right, so this is about the virtual lab. Apart from these, there is one assessment tool that is basically used for the assessment purpose, that is in the Kahoot. I hope that most of you have heard about it. It is like a game play, okay, and it is very useful tool, and it's like a fun. So today we are also going to use the Kahoot and just tell you how it is used and how it is useful in the environmental sciences. Okay, so that is a Kahoot, and because everyone having a mobile phones with you, and you can just open the Play Store and just download the Kahoot app. Okay, KAHOOT, Kahoot app. So today we are going to demonstrate you the one virtual lab and one Kahoot play. Okay, so I'm just on my screen and share the screen of the virtual lab. Just to give me a minute. Okay, so this is the screen. This is the website of virtual lab developed by the Ministry of Education under the National Mission of the Education. So all of you, please open this link that I have provided in the chat box. In this link, you can just see the environmental engineering lab first. In this lab, you can see that there are different types of experiment is also listed over here. And environmental engineering lab 2 is also there. You can see the list of experiments like determination of TDS, TSS, alkalinity, hardness, chloride, total iron, pH, and BOD. Okay. And in the environmental engineering lab 2, we also having a different type of other experiments like determination of chemical oxygen demand, LM coagulation, determination of break point fluorine demand in the water, sulfate in the water, fluoride in the water, dissolved oxygen in the water, and turbidity in the water. We just take the LM coagulation today. We'll see. So all of you, please open this link and open the LM coagulation, and just go with me. Just click on the theory part, and you can see that the theory of this experiment is given over here. It is nothing but just like a jar test. I hope that environmental science students have know about the jar test. Jar test is basically used to optimize the doses and the time of the alum that is basically given in a coagulation and the flocculation processes of the water and the wastewater treatment plants. Okay, so it is a jar test to know the optimum. What is the optimum dose of the alum that we are using, that we have to use in the coagulation and the flocculation processes. Okay, so to determine the optimum dose, we basically do the jar test. The pre-test is also there. Okay, some questions is also there. You can do these questions also. Procedure of the experiment is also there. So all of you, please open this experiment, and we do the simulation of this experiment. Okay, so alum coagulation, the objective is to determine the optimum alum dose for the water and the wastewater treatment by the jar test. And apparatus used is a nephrometer, nephrometric tubes, pipette, beaker, stirrer, and measuring cylinder. Okay, and solution here we are using the alum solution, that is the Al2SO4 thrice 18H2O, aluminum sulfate. Okay, so this is our jar test experiment. First of all, you just take a 50 ml of the water sample. Just click on the whatever the arrow is showing, just click on that point only. Okay, so first of all, you just take the 50 ml of the water sample in the 6 beakers using the measuring cylinder. You use the measuring cylinder and pour this water sample in a beaker, 6 beakers. Similarly, we just take a 6 beaker like there. Okay, and after that, you just add a different doses of the alum solution into the 6 beaker with the help of pipette, with 1 as a blank. Okay, so for 1, we just not adding anything, and in the rest of the 5, we just add the alum solution. Okay, and in an increasing order. For example, in the first beaker, you just add 0 ml, the second beaker just 1 ml, and the second one 2 ml, 3 ml, 4 ml, like that. Please do along with me, all of you. Please do along with me. In each and every beaker, you have to add the alum solution, that is the aluminum sulfate. And in between, there is some question is also there, like unit of the turbidity is the NTU. What is the NTU stand for? Nephrometric turbidity unit. Okay, so this is a jar test experiment setup, and you need to put each and every beaker under the mixture stirrer. Okay, and keep the remaining 5 beaker under the approximate stirrer, and stir the mixture approximate. So you just put the stirrer under the beaker, in the beaker, and start the instruments. First of all, you need to start it with a slow RPM, a low RPM, because flocculation takes place, coagulation takes place. And after that, the slow RPM, the less RPM, first of all, the the more RPM, and then the less RPM. Because first we need to mix the alum in a water, and after that, when the alum is completely mixed in the water, and then flocks will become formed, that is known as percolations, and that is basically occur at a less RPM. First of all, set the RPM like 100 RPM and start the system. And after just less down the RPM and start the flocculator also, Jarter separator. So that is basically Jarter separator apparatus in which we mix the our coagulant and allow them to flocculate at a lesser RPM, that is the 30 RPM for a 20 minutes. First, we just do this experiment at the less RPM, for the more RPM for a few minutes, and after that the less RPM at the 20 minutes. And after that, the instrument will automatically close, and you need to pull out the stirrer and take out the beaker. Remaining 5 beakers will also take it out in the similar way, and allow the sample to settle down for the 2 or 3 minutes without disturbance, so that whatever the flocks is become forms will be settled down. And after that, you need to take nephelometric tube and fill it with the sample in a beaker, and similarly fill all the nephelometric tubes from the beaker. Place the cap of the nephelometric tube, and after that you need to calibrate your nephlometer. For that, we have the nephlometer. Place the sampling cell. Place the distilled water to make a nephlometer 0, and set the reading 0 by adjusting the chorus fine knobs of the fine knob. And after that, take out the distilled water and place your 100 NTU calibration standard in the sample holder of the nephlometer, and adjust the calibration knob to set the reading to 100. So this is a simple procedure of calibration of the nephlometer. And after that, you can directly put your sample. Nephlometer has the calibrated in order to eliminate reduce the bias of the reading over the all ranges. It is a true. And after that, you need to directly measure the turbidity of your sample. After the calibration, you just put your sample in the sampling cell of the nephlometer and take the reading. Whatever the reading is coming, that is your final reading of the sample. Like 60.5 is the sample reading of the sample. Similarly, we just take a reading of each and every nephlometric tube, 6 nephlometric tubes. Similarly, measure the turbidity of the remaining samples. Coagulation is a slow mixing technique which promotes agglomeration and allows particles to settle down. That is the correct answer. And after that, you need to put the value to the alum doses like 0 ml. So that is the mean 0. And after that 10, 20, 30, 40, and 50. So here you can see the result is shown in the form of graph. You can see one line graph is also there, and an x-axis optimum alum doses are there, and the y-axis turbidity will be there in milligram per liter. Turbidity should be in the NTU, okay, and optimum dose in a milligram per liter. It is wrongly written over here. Optimum alum dose you need to find out from this graph. Like optimum dose are this dose that is on which the turbidity is the minimum. The maximum clearance will be there on which reading we have just take the optimum dose. So minimum reading of the turbidity is at the 14.2. Okay, you can see 14.2 is your optimum dose, optimum alum dose in a milligram per liter. So we just write down the reading over here of 14.2 and check our reading. So that is true. That is right. So in this way, you can just see the experiment in the form of optimum dose. I hope that all of you have done this experiment at your place. And 14.2 is the optimum quantity of the alum required to give a sample with the turbidity 11.3 NTU. Okay. This is the inference of this experiment. Optimum quantity of the alum required for the given sample with the turbidity 11.3 NTU is 14.2 milligram per liter. Okay, I hope that you understand this experiment. Now we just do one small exercise on the assessment tool, that is the Kahoot. Okay, I just prepare a few questions in the form of Kahoot. All of you, please download the Kahoot app from the Google Play Store. Kahoot app, K-A-H-O-O-T. From the Android mobile phone, you can just download it. Or if you are open your web browser, then you can open the kahoot.it, K-A-H-O-O-T.it. Okay, in which you can see one game pin. Game pin is visible on the screen, 3353127. You just please give the game pin in the Kahoot app or website, kahoot.it, and write your name, and you can join. In this way, we just see how many participants are active in this FDP. Please do it fast. Your name will be displayed on the screen. Yeah, Akansha is joined. Very good. Akasha has joined. All of you, please just download the game Kahoot. There is one small app. You can download it from the Play Store, or you can just write down the kahoot.it in the web browser. You just open the Kahoot. Yeah, Maitri is also joined. Very good. And in this app, you can there is one is asking the game pin. You just write down the game pin 3353127. Okay, and when the question is shown on the screen, and when I start this app, this Kahoot quiz, then the question is, you can see on the screen, on the screen of the Zoom platform, and all having 4 options, and this options is also having a coloring code, and only the coloring code is available on your mobile app. So you just see the answer of the question on the Zoom app, and based on the right color, you just click on the right color on your mobile app. The question is not shown on the mobile phone or kahoot.it web browser. Question you can see on the mobile. So only 2 students only joined. Why are not others? Please download the app and join. All of you. Only 2 are active participants only. I need more participants. All students, please join. All PhD students, all research scholars, all PG, UG students, please join. Please open the web browser kahoot.it, or you can, if you have a mobile phone, then go to the Google Play Store and download the app kahoot.it. It is a very small app. Quickly download it. Yes, students, please join. I'm just waiting for a few minutes only, and then we'll start. Okay, so rest of the students, if you are taking a time in downloading the app, you can just, when your app is become downloaded, then just click, just give the game pin 3353127. This game pin is always be available on the screen. Okay, we just start the quiz now. So you can see the question on the screen, and this question is having a four options. Each option is having a color code, and color code is visible on your mobile phones. Okay, so rest of the students can join the game pin 3353127. That is available written over here. Those students who joined, please answer. You can open the web browser. You can open the web browser kahoot.it. Kahoot.it. If you are not able to download the app, just please open this website kahoot.it. KASOOT.it. From this website, you can join. All research scholars, please join. Okay, both the students have give the right answer. Then I just click on the next question. Very good. And the beauty of this app is this, this game is that more quickly you answer, the more points you will get. The more quick you answer, the more points you get. So this is the question, and these are the answers. These are the options. Please open the kahoot.it in the web browser or download the app. All the research scholars, all the UG and PG students. Only 2 students have joined. Okay, one more student have joined. Okay, Manisha, I joined. All right, good. So other research scholars, please also join. It's a simple questions in environmental sciences. I request to all the students, all the participants to please join. You can download the Kahoot app from the Google Play Store, or you can just type the kahoot.it in the web browser. You can able to join. And then after the joining, just give the game pin 3353127. I just repeat 3353127. Please answer the question by clicking the color code of the options. Okay, the coloring code is available, is seen, you can mobile app or the web browser. Based on the color options, you can just choose the color options only. Okay, one more students have joined. Good. So you can see the question on the screen, on the Zoom screen. You can see the question on the Zoom screen, and you can answer the question. You can answer the question by clicking the colors or coloring options. The option is not visible on your mobile screen or your web browser. Only the color options is available. Only colors are available. So based on the correct color, you can answer. Click on the correct color of the options. Very good. I just request for the students also to please join the quiz. You can just download the kahoot.it. You can just download the Kahoot app from the Google Play Store, or you can write the kahoot.it on the web browser. And game pin is the 3353127. It is written over here in the lower side of this screen. You can see the game pin is also written over here, 3353127. The rest of the students, please answer. Very good. Now all the students have answered. Other participants will also please join, apart from the students. Research scholars and other participants, please join the quiz. You can download the Kahoot app on the Google Play Store, or if you're not able to download the Kahoot app on the Google Play Store, you can just write the kahoot.it on the web browser. You can see from this kahoot.it, you just and the Kahoot app, just give the game pin 3353127. You can able to join the quiz or the game. All the UG and PG students, MSc, BSc students, please join. All the participants, please join. Research scholars, faculty members, please join the quiz. The game pin is the 3353127. More quick you answer, the more points you get. Other participants also answered, so that the question will be closed quickly. If all the participants answer, then question is closed quickly. Good. Four more questions. Three participants answered. Good. All participants answered right. Name of the top 4 or 5 participants are shown on the screen after the question, and their marks is also shown on over here. This is the second last question. This is the last question. Okay, so this is the last question. Now let's show the results. On the third position, Manisha, then second Moitri, and the fourth position, Akansha is there. Okay. Okay, so this is how we can just play Kahoot app. Okay. So, okay, thank you so much. Over to you, Charu.
Thank you, sir. Sir, we have some questions from our participants. So, sir, with your permission, we can take up the
Yes, yes.
questions.
Yes, yes, please.
The first question is, what are the limitations of virtual labs in environmental sciences?
The limitation is that you cannot do the experiment physically. The limitation is that, and after that, I know some some calibrated standards cannot be, some calibrated things cannot be done. Like some sophisticated instruments cannot be tried on the virtual lab. For example, if you want to do the ICP-MS or the ICAAS, that cannot be happen possible on the virtual lab. You can do the small experiment that can be simulated virtually, but in future, if it is possible, then that can also be takes place. So that is the limitations, and for then the physical form of the experiment you cannot do. You just do it experiment at a particular level. Then the true value cannot be available. Okay. So for example, if you want to do the sample of the river, then that sample cannot be do on the virtual level. But virtually you can just use it for the teaching purposes, basically.
The second question is, how is ICT used for environmental conservation more sustainably?
Okay, so basically ICT can use in the environmental sciences. For example, if you want to use the artificial intelligence, then you can just predict the water quality or predict the air quality, and then in that way you can just conserve the environment sustainably. And if you want to use the lux meter or the sound meter app, then you can just see what is the sound level. Then you can just try to reduce the sound level. Then in that way you can conserve the environment sustainably.
Okay, sir.
Yes.
So third question is, please clarify which standards are using in virtual labs.
In virtual lab, the standards are also virtually calibrated. Okay, so we just do the things in the simulation way. We just pre-calibrated standards are there. It's having a numerical value. Okay, it's not a true standards. It's a virtually standards is used. And if the instruments become simulated, then the standard is also simulated. It gives only the reading. That is the virtual only. Okay, it's not a true.
Okay, sir. Thank you, sir, for taking out time for your busy schedule, and for our participants. It was truly a privilege to hear from you. Thank you, sir.
Thank you. Thank you, Charu.
Moving ahead, the next dignitary to honor us with his presence is Dr. Vinay Kumar Khanna. Dr. Khanna is the Chief Scientist in the Regulatory Toxicology Group at CSIR, Indian Institute of Toxicological Research, Lucknow. Sir, I now request you to please address the gathering with your talk on the topic, Novel Strategies for Low-Grain Arsenic Rice Crop for Sustainable Environment and Agriculture. But before that, we would like to facilitate with you virtual sapling. Dr. Renu Thupar is doing the honors. Welcome, sir. Over to you.
Hi. Charu, I'd like to share the slides.
Yes, sir, you can share the slides.
Just a moment. I'm just trying to get
Okay,
these
sir.
slides on the screen. Just this will take few seconds now.
Okay, sir.
Can you see the slides?
Yes, sir.
Are my slides on the screen itself, Charu?
Yes, sir.
Okay. So very good evening to all of you. And at the outset, I would like to thank my colleague, Dr. V.P. Sharma and other colleagues at Amity for giving me this opportunity to be part of this program. And also, before I share some of the ideas for using the quercetin, I also like to apologize for not being with you yesterday because I was occupied in one of the meetings. And again, I like to keep my talk also very short because I have to attend another meetings also. So, but nevertheless, I would like to share some of the ideas that as a part of the research that we are carrying out on quercetin, for its use as a neuroprotectant in chemical-induced neurotoxicity and also in certain neurodegenerative diseases. You know, quercetin is one of the bioflavonoid which is extensively present in vegetables, fruits, and also in some of the dietary products, especially in the green tea. And high levels of quercetin have been found to be present in apple and berries. Besides this, the quercetin is also reported to be present in certain medicinal plants, especially in ginkgo biloba and a couple of other medicinal plants. And this bioflavonoid has been found to have a wide pharmacological spectrum, which is quite which has fascinated quite a lot to the scientists, because it has been found to have the anti-inflammatory activity, because quercetin modulates the levels of the pro-inflammatory and the cytokines which are involved in the pro-inflammatory and also in the anti-inflammatory process. It also modulates a couple of other pathways, especially which is involved in the cyclooxygenase and also in the lipoxygenase pathway. If we talk about the antioxidant potential of quercetin as a part of its pharmacological spectrum, it has amused quite a lot because it scavenges the free radical species which are generated, and also it has a lot of antioxidant properties. And because of this anti-inflammatory and antioxidant properties, which are suitably attributed in the neurological conditions and also in the neurotoxicity conditions, it has been adequately utilized to assess the protective and therapeutic effect of quercetin in the disease condition. Besides that, it also has been found to modulate the the apoptotic pathway, because it modulates the proteins which are involved, especially the BAX, BCL2, and also the scooter protein caspase 3, which are involved in the process of the apoptosis. And if we talk about the neurotrophic effects of quercetin, it has been found to modulate the effect of the BDNF, which is known as the brain-derived neurotrophic factor, and also the neurotrophic factor, other neurotrophic factors, and also the TEP, which is very important to modulate the signaling pathways. Besides that, quercetin also has the metal chelating property. Therefore, it is also being used as one of the chelator in certain processes. As I just now mentioned, that the anti-inflammatory and the antioxidant potential of quercetin has fascinated many of the scientists just to assess its therapeutic and protective potential in disease condition. A number of preclinical studies have been carried out using different models of disease conditions, wherein it has been found, especially in the cases of Parkinson's disease, where people have used different models, right from the 6-OHDA to rotenone-induced model, and even the MPTP, where, as you will appreciate, that in case of Parkinson's disease, the degeneration of dopaminergic neurons in the substantia nigra that leads to deficiency of dopamine in the striatum is one of the important mechanism that is involved in this process. And treatment with dopamine in the disease model of Parkinson disease has been found to correct the levels of dopamine and also reduce the level of the alpha-synuclein, which is one of the aggregatory protein, which is found to be present and also one of the causative factor in the case of this neurodegenerative disease. Besides, in the case of the Alzheimer's disease, where a number of experimental models, right from the scopolamine to other chemical-induced models have been used, where quercetin has been found to decrease the senile plaques, which are normally found because of the amyloid precursor proteins and also the neurofibrillary tangles, which are found at the level of the hippocampus, which is an important area of the brain that controls the learning and memory. Besides, the learning and memory, which was found to be impaired in the disease condition, especially in the AD, were also found to be corrected, especially in the cases when the rats were treated with quercetin. Taking up the advantage of the pharmacological spectrum of quercetin, people have also tried to assess its protective potential in animal model of Huntington disease. This is another spectrum of disease which also involves the dopaminergic pathway. And as a matter of fact, the basal ganglia is really involved in it. And during the process, the striatum is affected, wherein the increased level of the GABAergic receptors, they are found to be affected during this process, and it also leads to increase in the levels of dopamine. And during the course of the disease process, the mitochondrial integrity is also affected. So what we found that, and number of studies have also demonstrated that simultaneous treatment with quercetin was found to protect these changes and also to maintain the integrity of the mitochondria. In the case of ALS, that is amyotrophic lateral sclerosis, which is another neurodegenerative disease, wherein...
Excuse me, sir.
Sorry?
So your slides are not moving.
I'm still speaking on the same slide, right?
Okay, okay, sir. Sorry, sir.
This is on the amyotrophic lateral sclerosis, wherein a number of studies have found that quercetin could protect the chemical-induced changes, especially in the animal models of ALS. Another spectrum of disease that is especially in the stress-induced anxiety and depression, where quercetin has been found to protect the cholinergic and dopaminergic circuitry. Besides that, it was also found to correct the expression of the NMDA receptors and also the levels of the NOS, which are found to be altered in the disease process. So with the demonstration, also the process, while we have found that quercetin crosses the blood-brain barrier, there has been further interest to understand whether it could be used as a neuroprotectant. So based on that, I'd like to spend a minute here just on this particular slide, that as I had mentioned, that different models of cognitive deficits have been developed to understand the mechanism and also to assess the potential of quercetin, wherein people have used the streptozotocin-induced memory, the mouse model and rat model of memory impairment, and is where they found that the increased oxidative stress was found to be corrected, as can be seen here, that treatment with quercetin was found to decrease the levels of malonyl dialdehyde nitrite levels, and also it was found to correct the acetylcholinesterase activity. It also was found to enhance the levels of glutathione. Therefore, the strong antioxidant potential, that's what I had mentioned in the beginning, was found to be one of the important contributor to protect these changes. In another study that was carried out in the amyloid beta-induced mouse model of AD disease, wherein increased oxidative stress, as I had mentioned right now, was also found to be protected in this model, wherein I had mentioned in one of the slides earlier that the inflammatory pathway.
Excuse me, sir.
Sorry.
Sir, we can see your first slide only. We can see the very first slide of your presentation only.
So these slides are not moving, is it?
No, sir.
I'll just try once again. I'll again load the slides. Can you see the slide right now?
Yes, sir.
Now, are the slides moving now?
Yes, sir.
We can see the quercetin pharmacological spectrum and the preclinical leads.
This is the second slide now. Can you see the slide now?
No, sir. Sir, actually, it is not on slide mode, so it is not changing.
It is on slide mode because
So you have to either change it through.
just give me a few seconds.
Yes, sir.
I've asked my IT person. Yes, come here. Can you see the slide right now, or is it not moving right?
Sir, it is. We can see the slide, but it is not on the presentation mode. We can see the column at extreme right hand side. So it's not on the presentation mode. So either you have to change it through the column only, or otherwise, if you can put it on slide mode, then through the arrow keys, it can be moved. Yes, sir, it's there now.
Okay.
So can you just check it once whether it is? Yes, sir. Perfect, perfect, sir. It's moving now.
Fine, thank you. I'm so sorry for this confusion.
It's okay, sir. Please.
So just in the interest of time, I will not go into the detail once again, but I like to share some of the things and the findings which have interested, which will be of interest to many of us. That the animal models which have been developed as far as the memory impairment and cognitive deficits, people have used different animal models of using different drugs and chemicals, especially streptozotocin induced. That's what I have mentioned. And also the amyloid beta induced mouse model of AD disease, where the role of inflammation was found to be there, and quercetin was found to protect these changes by modulating the levels of NF-alpha beta and JAK-STAT pathway. Besides that, it also was found to modulate the levels of P65 and MAPK signaling. It also was found to modulate effectively the levels of one of the neurotrophic factors, that is the brain-derived neurotrophic factor, and also modulate the signaling other targets which are involved in the signaling. In the scopolamine-induced rat model of memory impairment, treatment with quercetin was found to decrease the beta amyloid level, which has been found to be one of the important contributors in the disease process, and also it enhance the levels of GSH and other targets which enhance the levels of the oxidative stress. In the mouse model of D-galactose-induced aged animals, it was found to effectively modulate the NRF2-ARE signaling, which is the antioxidant response element, which is strongly contribute in the antioxidant response process. And besides that, it also could effectively control the cell apoptosis. So taking the advantage of this information, we further developed an animal model of cadmium-induced cognitive deficits, and because these are the studies which we are pursuing here at IITR for last many years, and cadmium is one of the trace elements which is extensively present as a trace element which is present in the environment, and it has extensive anthropogenic uses, both in the mining and other industries. And it also, on the cadmium is also used as pigment in the paint and plastic industries. Besides, it also has extensive uses in the nickel cadmium batteries, and it is also preferred in the for the electroplating because of its non-corrosive nature. Besides that, cadmium salts are also used in the fertilizers and also has extensive anthropogenic uses. Therefore, human exposure due to environmental exposure to cadmium is quite imminent. Besides the environment, this occupational exposure, high levels of cadmium have also been found to be present in the drinking water, in seafood, and also in the crops which are grown in cadmium contaminated soil. And tobacco smoke, of course, is one of the very strong contributor, which enhances the risk of normal population. basically. So human exposure to cadmium could occur both under occupational and non-occupational settings. And moreover, the fact cadmium, in spite of the fact cadmium enhances the risk of cancers, and in human studies, it has been found that increased levels of cadmium enhances the risk to develop hypertension and also the cardiovascular diseases. Besides, it also enhances the risk of cirrhosis, and of course, the kidney is one of the important targets. As cadmium competes with calcium, so what happens in the case of in certain situations, while it replaces the calcium, it causes or enhances the risk of osteoporosis. So of various organs in the body, brain is definitely one of the soft target of the toxic effects of cadmium. And in this situation, because brain has very high levels of PUFA, which is referred normally as polyunsaturated fatty acids, and also is deficient in the antioxidant system. Therefore, enhanced oxidative stress in certain situations is one of the important mechanism which is largely accepted in the case of the neurodegenerative diseases. Besides, in the interest of time, I will not go into the details of the epidemiological and environmental studies, which have very well correlated that cadmium enhances the risk of cognitive deficits. So with that, taking the advantage of that situation, we have developed an animal rat model of cadmium-induced cognitive deficit, wherein we treated the rats with cadmium, and in an attempt to identify the targets which could be involved and associated in cadmium-induced cognitive deficits. And on the other hand, we also wanted to see whether quercetin has the potential to protect these cadmium-induced cognitive deficits. So here I summarized one of the studies that we carried out with the quercetin. As I had mentioned, that we are carrying out these studies for over last 10 years. On this is a long-term project that we are carrying out these studies. Here, one of the cholinergic integrity of the cholinergic signaling, especially in the hippocampus and frontal cortex, they are very important and they modulate the cognitive functions. So what we found here, treatment with the cadmium in rats was found to disrupt the cholinergic signaling, as can be seen here. If there is increase in the levels of the acetylcholine at the synapse, that will further affect the integrity of the cholinergic receptors. So what we did here, because out of the 5 different type of cholinergic muscarinic receptors in the hippocampus, M1, M2, and M4, they have been found to be associated to modulate learning and memory. So we carried out both the transcriptional and translational studies, wherein we found that the mRNA expression of M1, M2, and M4 was found to be decreased, and these changes were found to be consistent with the translation label. We further confirmed these findings by carrying out the binding assays for the cholinergic receptor or the cholinergic receptors, both in hippocampus and frontal cortex, wherein we used the 3H-QNV binding. And what we found here, that treatment with cadmium was found to affect the cholinergic receptors, which was found to contribute in these changes. This is PKC beta 1, which is a downstream target, and even the PKC beta 1, those rats which are devoid of PKC beta 1, they have been found to have the learning and memory deficits. What we found here in case of treatment with the cadmium was found to decrease the protein levels of PKC beta 1, which was very well correlated with the impairment in learning and memory. So once we found that cadmium disrupts the integrity of the cholinergic signaling, especially in frontal cortex and hippocampus, we further wanted to understand what could be the possible reason for these changes, as mitochondria is certainly important for the integrity of the cell. So we further focused on the mitochondria, because and then cadmium being pro-oxidant in nature, it was found to enhance the free radical generation and also impair the antioxidant mechanism, wherein it was found to enhance the levels enhance the apoptosis by modulating the pro-apoptotic those proteins which are involved in the pro-apoptotic and also especially BAX, wherein the protein levels of BAX were found to be increased, and the levels of BCL2, which is an anti-apoptotic protein, was found to be decreased. Caspase 3 activation was also found both in hippocampus and also in frontal cortex on treatment with cadmium. This is an important distributor protein, wherein we could suggest that cadmium probably enhanced generation of free radicals could possibly contribute in enhanced apoptosis. So after having studied the effect on the apoptotic proteins, we further wanted to see how this cadmium, whether it has any effect on the cell survival pathway, and especially in those proteins, especially in the JNK and P38 signaling, which is very important to modulate the cell signaling pathway. And we could demonstrate effectively that the levels of JNK1, JNK3, JNK1, 2, and 3 proteins is significantly affected on exposure to cadmium, which was further found to be consistent with the increase in the levels of P38. Based on that, we could further suggest that cadmium could disrupt the JNK signaling, which is very important to modulate the cell survival pathway. And this further contributes in the neuronal death. That we further confirmed by carrying out the histological and ultrasexual studies using transmission electron microscopy. So based on these studies, we could suggest that cadmium disrupts the cholinergic signaling, wherein it involves the mitochondrial pathway, which contribute in the disruption of the cholinergic signaling, and quercetin has the ability to attenuate cadmium-induced cholinergic deficits by modulating the oxidative stress, apoptosis, and functional activity of mitochondria. And based on that, we could further suggest that cadmium-induced cognitive deficits in rats were also found to be protected on simultaneous treatment with quercetin. So after having studied the effect on the cholinergic circuitry, our interest was once again on the NMDA receptors. And reason for carrying out these studies on the NMDA receptors was because of one of the reasons was that they are inotropic in nature and gated by glutamate and glycine. Besides that, they are a heteromeric complex of 4 subunits which are differentially distributed in the brain. And they modulate the learning and memory, especially the long-term potentiation and long-term depression, which are very effectively controlled by NMDA receptors. And role of NMDA receptors and its post-synaptic signaling is very well accepted to modulate in the learning and memory. So what we found here, the cadmium was found to disrupt the levels of NR2A and NR1 and NR2A receptor subunits. And these subunits, which are normally anchored on the postsynaptic density, that is PSD95 protein, that anchors this NMDA receptors, was also found to be effective. And during the course of the NMDA receptor signaling, CAMK2 alpha pathway is one of the important contributors that probably modulates the calcium signaling, which was also found to be the levels of CAMK2A were also found to be affected on treatment with cadmium. SYNGAP, which is another protein, which is also considered to be one of the negative regulators in this pathway, wherein we could further found that exposure to cadmium or treatment with cadmium, in other words, it enhances the levels of SYNGAP. And ultimately, we could also found that this TRK beta, which is one, and BDNF, which is one of the neurotrophic, for which the TRK beta is one of the important receptors. They are also affected on because the levels of BDNF associated with TRK beta were found to be decreased on treatment with quercetin or treatment with cadmium. However, simultaneous treatment with quercetin was found to protect these changes. Besides that, we could also demonstrate that this is another protein, that is PTEN, which is a tumor suppressor and involved in several diseases, especially cancers, diabetes, AD, and PD. And it negatively regulates the PI3K-AKT activity by exchanging the PIP3 to PIP2. And it also modulates the synaptic and cognitive functions in AD model. So based on that, we further wanted to see what is the effect of cadmium as such on the PTEN. What we found here, the level of the PTEN, as I have just now mentioned, it is also one of the negative regulators in the pathway. were found to be increased, which was further found to affect the other downstream targets, especially the GSK3 beta, on treatment with the cadmium, especially in hippocampus. Besides that, we further wanted to see, as I had mentioned in the beginning, that the ARE signaling that is very important because it is one of the key redox sensitive transcription factor, especially the NRF2, that regulates multiple antioxidants, and it binds to ARE, that is antioxidant response element, and induces expression of pro-survival proteins and cytoprotective enzymes. And decreased expression of this, especially the NRF2 and other ARNE signaling molecules, has been very well reported in neurodegenerative diseases and also in the condition of chemical induced neurotoxicity. So what we found here, that treatment with cadmium was found to decrease the level of NRF2 and also keep one, which is one of the excluded protein, which was further very well associated with the decrease in HO1, which is heme oxygenase 1. That is very important role in the as an antioxidant, the level of which were also found to be decreased. So based on this particular pathway, we could further suggest that the disruption in NMDA receptor and its downstream signaling on cadmium treatment was found to be associated with alterations in PTEN, AKT, and antioxidant response element signaling in hippocampus. And these changes that we observed were found to be protected by quercetin. So based on these studies, we could suggest this is not only the cholinergic signaling which is disrupted on treatment with cadmium, but this is also the NMDA receptor and its associated post-synaptic signaling is also disrupted, and probably these changes could be attributed because of cadmium-induced changes in the antioxidant response element binding, which further could modulate these changes in these signaling targets. We further wanted to understand the effect on the STEP, which is an critical enzyme that modulates the cognitive pathway, because alteration in the NMDA receptor that modulates STEP through a calcineurin and TP1 alpha pathway, and increased levels of STEP61 has been reported in neurodegenerative disease. Moreover, the fact because STEP modulates the expression of the NMDA receptors, that is the reason we further wanted to see what is the effect of cadmium as such on the level of the STEP. So what we found here, STEP is also one of the negative regulator. On treatment with cadmium was found to enhance the levels of STEP, and also it also disrupted its other targets, especially downstream, which are considered to be the substrates like PIK2, FIN, and PTP alpha on treatment with cadmium were found to be affected. And this cadmium-induced changes in the STEP and its targets were found to be protected on simultaneous treatment with quercetin. Based on that, we could further suggest that enhanced levels of STEP protein that resist synaptic strengthening by regulating the NMDA receptor signaling in hippocampus on cadmium treatment may be protected on simultaneous treatment with cadmium. So, after having discussed, I would like to spend 30 seconds on this particular slide, wherein we could further assess that quercetin has the potential to modulate the ERK, CREB, and BDNF signaling. Besides that, it also has the potential to modulate the NMDA receptor, and one of the studies where we have carried out is to further understand the effect on the AMPA receptors, and especially AMPK signaling is also affected, and it also modulates the NRF2 signaling and the JNK and RACE signaling, which is very effectively modulated by quercetin. After that, we also have carried out some studies to understand, because in case of cadmium, it also causes the dopaminergic dysfunctions, especially the motor dysfunctions, which are frequently reported in occupational workers which are exposed to cadmium. So in an attempt to understand what could be the possible mechanism, we could further demonstrate that cadmium disrupts the presynaptic signaling, especially targeting those proteins which are involved in the synthesis of dopamine, and it selectively targets the DAD2 receptors, because dopamine receptor is a very small family of 5 receptor types, and subtypes are there, and out of that DAD2 receptors were found to be target. But the interesting finding that we found that cadmium induced effect on the DAD2 receptors, it involves both the canonical pathway, wherein the PP1 alpha and CREB is there. And it also involves, on the other hand, it also involves a non-canonical pathway wherein it affects the GSK3 beta, and these changes were found to be associated with cadmium-induced motor deficits in rats. So these are the preclinical studies. That's what, in an attempt to identify the targets which are involved in the cadmium-induced cognitive deficits and also cadmium-induced motor deficits, wherein we could provide an evidence about the neurochemical alterations and also the signaling targets which are involved in the process. We also could demonstrate that how well quercetin is able to modulate these targets. A number of clinical studies are being carried out at many of the labs, wherein the effect of quercetin is being assessed in the clinical cases of AD and PD patients. And one of these studies where in a small group of AD patients, people use the 18 gram of onion powder, and they were probably treated in a small group of AD patients for 4 weeks, and this onion powder was found to contain 80 milligram of quercetin, a glycone, and these patients are found to have reported to have improved memory recall. However, in another community-based studies and 1,002 residents in West North Carolina, there was no significant effect on the memory was observed. In case of the PD patients, again, the consumption of red grape, blackcurrant, vegetables, juices, and wine was found to improve the motor activity. Bioavailability of quercetin is one of the important point to be taken into consideration because of the fact that quercetin is lipophilic in nature. So because it is lipophilic in nature, so it is not readily soluble in water. And when it is not readily soluble in water, therefore, availability of quercetin as a part of the metabolism in the circulation and also in the tissue is one of the important. While it crosses the blood-brain barrier, that is one of the very interesting things. However, the amount of quercetin to reach the brain and also into the target is an important thing, for which people are using different approaches, and they are also using the we are also using the nanotized formulation to understand if we are able to at all enhance the bioavailability of. So these are still the challenges because the poor solubility is what I mentioned, and it also has a short biological half-life and low bioavailability and poor permeability, where besides that, it also has a chemical instability. So the exact mechanism and targets which are modulated by quercetin need to be assessed well for its effective use as a neuroprotectant. The studies that I have shared with you, she is Richa Gupta. Richa is the faculty right now, and she was the one who started these studies, and especially those that I have shared with you on the cholinergic signaling. And Anugya is the one who further pursued these studies to identify the role of NMDA receptor and its post-synaptic signaling. She is just in the process to submit her thesis, and Anima also contributed to understand the role of ARE signaling. She is also in the process to submit her thesis. With these words, I would like to thank all of you for your patient hearing. And if there are any questions, please, I'll be very happy to respond to them. Thank you.
Thank you, sir, for such a detailed and enlightening session. Sir, we have few questions from participants. So, sir, with your permission, we can take up the questions.
Please.
So, first question is, how can we study the exact mechanism of action of a drug which believe it has a neuroprotective effect?
For that, we have to. Can you repeat the question once again?
Yes, sir. How can we study the exact mechanism of action of a drug which believe it has a neuroprotective effect?
For that, you have to assess the targets which are involved in the process. Only thing, once we identify the target, that is one thing, we will be able to understand the protective potential of that particular compound.
Okay, sir. So second question is, what is the role of neuroprotective drugs in Parkinson's disease?
This is a very interesting question. Somebody has, some of the colleague has asked. I must appreciate. This is one of the things which is
I think she's facing some network issue.
Amrina, I would like to say a word.
Yes, sir, please, please. Yes, sir.
Good afternoon. If there are no further questions, and if there are more and more questions with such a neurotoxicologist, we would take care. And I think MIT will take care that all the questions are forwarded to Dr. Vinay Khanna so that he can answer it. I would just say a word of thanks to Professor V.K. Khanna. He is not only a friend, but a well-known neurotoxicologist in the country. He has delivered several lectures at the various platforms. So I can say that today's participants are fortunate enough to hear such a wide experience lecture of Professor Vinay Khanna in such simple language. These are my words of appreciation from both the organizers, Amity as well as from IITR. That's all. Thank you.
Thank you, Amrina. Thank you.
Thank you, sir. Tripathi sir, welcome. Thank you for joining, sir. Charu, may you please introduce?
Can we just cross-check, because I think I cannot see Tripathi sir.
Tripathi sir is here, ma'am.
Acha, he's here. Okay. Yes.
Camera is also on.
Charu.
I think Charu is rejoining us because she's facing some technical issue. So I would request.
Can I introduce my guide? Can I take the opportunity to introduce my guide?
Richa. Absolutely. You know, the teachers have a very special and a very, very, great position in our lives. They are, big contributors in shaping our
Always,
future. So go ahead, Richa, please. Please.
Thank you. So I'll take the opportunity to introduce my mentor and my guide also. Dr. Rudradev Tripathi did his MSc in botany and obtained his PhD in 1979 from Gorakhpur University. And the thesis was entitled Assay of Higher Plants for Antifungal Antibiotics and Some Aspects of Mode of Action of Active Principle. After this,
great.
he had joined NBRI, and
Thank you so
he
much.
is currently working as a NASI Senior Scientist Platinum Jubilee Fellow at CSIR National Botanical Research Institute, Lucknow. He has worked as Emeritus Scientist and Emeritus Professor. He has worked as Chief Scientist and Professor ACSIR and in the field of Plant Ecology and Environmental Science Division at the CSIR NBRI, Lucknow. He worked in different capacities as scientist, senior scientist, principal scientist, and senior principal scientist. He was group leader of ecotoxicology and bioremediation for about a decade. Sir has had numerous and widely appreciated papers on the different topics. His work on arsenic is a stalwart in itself. He has been a fellow and has been awarded with so many awards. He was also conferred upon Vigyan Ratna Award and Saryu Ratna Award. He is our Guruji, and we are always looking forward to hearing from him. And it's a great opportunity again, sir, to be hearing from you today as well. We would like to felicitate you with a virtual Tulsi sapling. So please accept our virtual Tulsi sapling as has been our tradition to honor our guests.
Welcome Tripathi sir, welcome to MIT.
Sir, you may need to unmute yourself. Tripathi sir, we are unable to hear you.
Yeah, okay. Am I audible?
Yes, sir, very much. So may I share the slides from my side?
Yeah. Okay. Oh, okay. When I will say to change, only then you change.
Okay, sure, sir.
Yeah.
Thank you, sir.
So, can I start?
Yes, sir, please.
Okay. So I am very much grateful to the organizers, especially Dr. Richa Dave, who has been my student, and I had a great pleasure of working with her for many years. And it's really a very pleasant time for me to give a presentation in a training program organized by the MIT University and my sister laboratory, Indian Institute of Toxicological Research. So it's really a very pleasant opportunity for me to give a presentation. And thank you, Richa, for a very big introduction. So the topic which I have chosen today is the novel strategies for reducing arsenic level in rice crop for sustainable environment and agriculture. And this topic was initiated by my students like Richa Dave, and then it is quite some time, and we could do something. So I will share some of my experiences done in this area. Next, please. So the arsenic is a carcinogen, and it's a non-threshold carcinogen, and it is responsible for causing many diseases, and the arsenic is present in the rocks, and due to the chemical withering and this reduction process, the inorganic arsenic is released into the groundwater. And as we know that this groundwater is mainly used for the irrigation of the crops. And most of the cereal crops, they require a lot of water, but the rice or the paddy is one of the most water intensive crop, and it requires maximum water. And it is because of this that the arsenic which is present in the groundwater through some of the transporters present in the rice tissue, it gets entry into the rice plant and then into the grain. And many cattle consuming contaminated feedback indirectly contaminate the milk and other products. So it contaminates the whole whole food chain. And there's a hospital in the Patna, and there's a new hospital. I think it got open a decade back or so. So most of the cancer patients coming to that hospital, they are showing the symptoms of the arsenic-induced cancers. Next, please. So you can see some of these symptoms of this exposure of arsenic through groundwater. And you can see the different kind of hyperkeratosis on the soul. The children are affected, and in fact, the chicken meat is also contains arsenic. In the chicken, in the poultry, to increase the weight of the chicken, some arsenical compounds are being used. So the chicken weight gain is very high, but it is because of this they get into the exposure of the arsenic. So even the chicken in the McDonald's is also contaminated with arsenic. And there's some of the another route of getting the arsenic into the food chain is rice. So one is because of the exposure of the arsenic from the water, and according to World Health Organization, the maximum permissible limit of arsenic in the water is 10 microgram per liter or 10 ppb. And the other route of exposure is the rice consumption. And because of some of the transporters like phosphate transporters and aquaporin transporters, these arsenic gets in the rice plant, and then it's get into the grains of the arsenic. And the maximum permissible limit of arsenic, the human body is 2 microgram per kg. So we are not permitted to take anything beyond 2 microgram per kg of the body weight. So most of the drugs are taken on the body weight basis, and so some of these arsenic is also, its level is fixed on the body weight basis. Next, please. Next. So this is a survey map where we have taken the groundwater contamination of the arsenic. So you can see that these are the union territories, these are the different states, and there are 3 levels, A, B, C is written here. So so these are you can see that these are the union territories there, and these are the states. Then there are A, B, C numbers. A is the number of samples, B is the maximum permissible and maximum arsenic level in the groundwater, and C is the percentage of the samples which are having their the contamination level more than more than 50 microgram per liter. So you can see that in the most contaminated the state, the West Bengal, and the Bangladesh is the most contaminated with arsenic, and the hottest spot. So in fact, the West Bengal and Bangladesh are they are the joint, and they are together. And in fact, we have the maximum problem in the West Bengal. You can see as high as 3,700 microgram per liter arsenic is there at some places because the number of samples were very high. So the highest sample was having 3,700 microgram per liter. You can see that in Bihar, you have a very high level of arsenic. Then in the Chhattisgarh, you have 45-70 microgram per liter of arsenic in some samples against the 10 microgram per liter maximum permissible limit given by the World Health Organization. So then you can see in Jharkhand also there are 1150 microgram per liter of arsenic. So you can see this, all these states have these arsenic level more than 50 microgram per liter, which is very high. And so you can see these northeastern states, and you can find that in Meghalaya you have 936 microgram per liter arsenic. So in the northeastern states also, in Assam it is 667 microgram per liter. So you can see that all these states we have mapped into this map. They contain very, very high level of arsenic, and from there it goes into the crops, and from the consumption of these grains, it contaminates the food chain, it goes to the human body. Next, please. So we have there was an urgent need to develop some rice variety which can be because it is not possible to clean the groundwater contamination of the river. You understand that this the surface water from the river water and pond water, they are not contaminated with arsenic, but the irrigation water, where the groundwater consumption is involved, or the drinking water in the villages where there is involved groundwater consumption involved, they are highly contaminated. So there was an urgent need of the arsenic variety which can contain the safer level of arsenic into the grains. So we had taken about 300 rice germ plant extractions from different states, and we had done some collaboration with the Rice Research Institute in the West Bengal, which is situated in the Hooghly district, and we have found that we have screened about 300 rice germ plants, and these were the trials were conducted at 6 different places, and all of these 6 different places in West Bengal where this rice cultivation was done for screening purposes. These are Chinsura, Durgapur, Purvasthali, Beldanga, Birnagar, Gaighata. And these Beldanga, Birnagar, and Gaighata, they were the most contaminated places, and the soil contamination, because of repeated irrigation, the soil gets contaminated. That corruption was above from 18 to 25 parts per million of arsenic was there. So these all blocks in the West Bengal, which are seen here, they are having the levels more than prescribed by WHO. So these are some of the 300 varieties which were screened. Next, please. So these are the different species of arsenic. From arsenic and arsenite, these are contaminated the groundwater. They are contaminated. They contaminate the land and the fields, and there is because of the presence of the bacteria, there is a challenger pathway which goes on in the microbes. It doesn't go in the plants, but it goes in the microbes, and arsenic get methylated because of this pathway, and because of the arsenic methyl transferase enzyme. So these are the different DMA, TMAO, TEMA, and these are the products of the arsenic methylation pathway. And this pathway is prevalent in the microorganisms. We've tried to make transgenic crops using these microbial gene. And these are some of the sugar derivative of the arsenic, and they are found in the marine organisms, in some arthropods also. And they just very the arsenate gets reduced to arsenite because of the arsenate reactant enzyme using glutathione as a coenzyme, taking electrons from the glutathione, and then it gets methylated by arsenic methyl transferase, and the methyl group of the methionine gets transferred, and this OH group of the arsenite gets methylated to arsenite. So in the process, the arsenic gets oxidized to arsenate 5th, and then again it is reduced to monomethyl arsenite. And once it is reduced, again it is methylated, and methylated products are oxidized, so it is again DMA5. Again it is reduced to DMA3, and it is again oxidized to arsenate trimethyl arsenium oxide, which is reduced to trimethyl arsine. So it's a gaseous form of arsenic, and it is because of these form the advantage we can make the transgenic plants, and by use we are using the CRISPR-Cas technology also to make the mutant plants which may be having very low arsenic level. So in the next another slides, I will tell how we have used this enzyme to make the transgenic plants, which can take up arsenic, but they can methylate it to the volatile forms, and therefore have the very safe level of arsenic in the grains. We have the next slide. Next slide, please. Can you change the slides? Dr. Richa, can you change the slide? Okay. Okay, so this was how the arsenic gets into the through NIP and then aquaporins. The arsenite gets into the roots, and from roots and then through phosphate transporters arsenate can enter, and arsenate gets reduced to arsenite, and arsenite can make complexes with the phytokeratin type of arsenic detoxifying peptides, and then through ABCC transporters it can be loaded into the vehicles. So this is here, this becomes immobile, and it is the enzyme phytokeratin synthase which produces the phytokeratins, and phytokeratins are the cysteine-rich metabolites. Dr. Richa has worked on these aspects, and through these complexes, arsenic gets complex, and then it is through ABCC kind of transporters, it can be sequestered into the immobile forms or non-mobile forms. And there are another kind of genes, the ACR13 genes, which can directly put arsenite into the vacuoles. And these genes are present only in the lower plants, in some pteridophytes, and they are not present in the rice or the higher plants. So the advantage of this gene is that they can directly transport it to the vacuoles. There is no need of any phytochelatin type of mediator. So once it's get into the roots through NIP and through the LSI2 type of transporter, it is effluxed into the xylem. Once get into the xylem, arsenic can go through the water stream. It can go into the upper story of the plants and can go to the shoot, and through inositol kind of transporter, which transports some sugars, it can be transported into the phloem. From phloem, it can go to the seeds, and it can contaminate the grains. So it's a filial tissue where arsenic can be loaded, and it is the mechanism how arsenic, which was in the soil or in the groundwater, gets contaminated into the food chain, and it's get contaminated the seeds and the grains. Next, please. I think we have. I was looking for this slide. So you can see that we had screened the 300 rice genotypes, and we found that these 7 rice genotypes, they are the low accumulating. And this is based on a formula, and that a 60 kg person can consume a rice variety which has 286 lower than 286 microgram per kg of the arsenic per kg of rice. So and again, this is based on a 60 kg body weight person can consume 420 gram of rice per day. I would like to tell you that in the state like West Bengal, they have their breakfast starts with rice, then their lunch in the rice based, and the dinner they have the rice based dinner. So thus their subsistence based diet, and in all the rice they consume more than 420 gram of rice. So you can see that one of the varieties CN1794-2-CSRN variety, which was developed, it was from the selection of a kernel type variety, which was genetically different, and we could make a selection based on the high productivity, more than 5 ton per hectare, and you can see that the arsenic level is very safe, less than 286 microgram per kg. So it's a safe rice variety, and this variety was released for cultivation in the arsenic contaminated area of West Bengal in 2016, and so West Bengal government approved this variety for cultivation in the arsenic contaminated regions, and then in 2019 this variety was approved by the Central Variety Release Committee of the ICR in the Krishi Bhavan. And so this variety is the variety which is cultivated by the farmers in the arsenic contaminated regions. So this variety may work as a gene resource to develop and breed other varieties. So these all varieties, the other varieties, Dynamoni CN1794-2-3-5, these all are the low arsenic content varieties which were identified after a screening program of the large number of varieties. And this is a variety which was developed as low arsenic variety, and this variety is safe for consumption. And okay, next please. Okay, you can see how this variety This was named as Muktasri. The Chief Minister of the Bengal gave this name to this variety. And this variety was developed from NBRI and Rice Research Institute joint collaborative work. And this is how it looks like this in the field. You see, there's very good variety, little long type. It's some kind of a basmati type variety. These are other popular varieties which are grown in the state of UP. Bengal Juhi, MTU 101, NTU 7029, again contain low grain of arsenic. Next, please. So you can see that this variety, which was developed as a low grade arsenic variety, it contains, you see this number 2 variety, number 2 bar in these are the 6, I told the 6 places, arsenic containing places, sites. And you see these yellow bars, there is a variety BRG12. So it accumulates very high amount of arsenic. So in all the trials, we keep this variety as a reference variety. So you can see that it contains very high level of arsenic. And this number 2 variety is the Muktasri variety, which was developed by NBRI and Rice Research Institute, Chinsura. So just look at the 2 numbers. So it contains very less arsenic. One of another good feature of this variety was while it was, you see this number 2, it is circled by the red circle. Here also it is circled by the. So this is the iron level. So there is a correlation with the variety which contains low arsenic. It contains very high level of arsenic, iron. So you can see iron in this one in the Kharif season, that is in the Rabi season in the Kharif season. So most of the productive varieties of rice, especially in the state of West Bengal or the rice growing states, you'll find they are sown in the Boro season, the dry season. The dry season requires more water. So in the more water means more arsenic contaminated water. So you can see the good performance of this variety when they are grown in the summer season. They have very low level of arsenic. At the same time, they have a very high level of iron. So the variety is very advantageous for people who are suffering with the malnutrition, especially the ladies and the children. So this variety is getting popularized, and it is grown by more than 135 farmers in that state of West Bengal. And we are trying to promote it in the other states also. Now the next, please. Next, please. So this variety Muktasri was developed because of the conventional breeding and selection. The another approach which we try to make is using the transgenic plants. And I'm very happy to tell you that Richa Dave worked on these in this plant, and we had very good publication on this transgenic plant, and in which he had used a phytoglandin synthase gene from an aquatic plant, Ceratophyllum diversum. Ceratophyllum diversum, it contains about 20, more than 20 phytoclatin variants which can complex with the arsenic. So this is ESR and the ICP-MS spectra, and we have isolated one of my students who is recently working as an assistant professor in Lucknow University. She did this work, and she identified these 14 variants of phytoclatin which can complex with this present in this Ceratophyllum diversum aquatic plant. So we have used the phytoclatin synthase of this gene and transformed the rice plant. So these phytoclatin synthase has 502 amino acid. It was about 17.5 1.75 kb long gene. Next, please. So we have used these genes and transformed the rice. These are the construct of the gene, and we have 35 S promoter we have used, and we have selected about 21. Finally, we selected 21 callas representing 7 independent rice lines, which were confirmed are the phytokeratin synthase gene. So, because of expression of overexpression of these transgenic rice plants synthesize more phytokinetics, which could make complexes with the arsenic and make the arsenic which was going to the rice plant and untoxic. Next, please. So you can see that these are the 7 different rice lines which were finally selected, and one line, the species 3 line was very effective because because you use the this Calliphal mosaic virus promoter, so phytokeratin are more accumulated in the roots, and because of that, more about 327% arsenic was detoxified in the root, and very less there was 59% inhibition of the arsenic in the grains of the transgenic rice plants. And these are transgenic, there was about 60% inhibition of the arsenic in the grain. And this variety was very good in the regard the seed output. So you can see the 1,000 weight seeds of the 1,000 weight, and this variety was having the maximum seed weight. So the production wise, while it was accumulating low grain arsenic, it was having more production in terms of the seed output and in terms of the productivity, about more than 5.5 tons per hectare. Next, please. So as I showed you in one of the slides, we identified one microorganism, a fungus, Vesta Dicala ornatacea, from the arsenic contaminated soils of the West Bengal, and this is a construct of this gene. This gene was 1.2 kb, so it was a short gene. And this gene was, when expressed in the plants, it produced a protein, which was an enzyme, and it was known as arsenite methyl transferase. And this enzyme, what it converts the arsenite into the trimethyl arsine, which is a volatile product. And I would like to tell you that this is a very good strategy for creating the transgenic phytoremediation also. So we made the tobacco plants, which were very efficient in the phytoremediation of arsenic. So when it was expressed in the rice plant, you can see that this is the non-transgenic plant, and this is the arsenic which was volatilizing. It is getting converted into trimethyl arsine, and these are the transgenic plants. So you can see more than 18-fold volatilization potential when it was exposed to 250 micromolar of arsenic or arsenite, and you can see that this is more than 50-fold arsenic is getting volatilized. So it's a very nice strategy, and Pankaj Verma and Shikha Verma, they did their PhD on this work, and we made the transgenic rice plants also. May I have the next slide? Next slide, please. Okay, so the rice transgenics were made, and you can see that these ricegenic plants, these are the non-transgenic plants. These are the L1, L2, L3 are the transgenic lines. So you can see that the plants they're untreated. They were all were plants like growing like that. But then we are treated with arsenite or arsenic, arsenite or arsenate. You can see that these non-transgenic plants were showing very high growth reduction in terms of the fresh weight, in terms of the root length or shoot length. You can see these are the wild plants. These are the wild plants, and they are non-transgenic. So they can see the transgenic plants are very long. This again the non-transgenic plants, very short plants, because arsenic was very toxic to them. And these transgenic plants could convert the inorganic arsenite into the trimethyl arsine. This escaped through the stomata, and the plants could tolerate the toxicity of the arsenic. So next, please. And you can see we measured the arsenic level in the grain also. So if this is in the root and shoot, you can see that's the transgenic L1, L2 they have the lower level of arsenic as compared to the wild type. You can see here about 50% reduction in the arsenic level. But the grain or the seeds which is consumed, you can see that is the unpolished seeds when exposed to arsenite, and when this is the polished seeds. So you can see that the arsenic level is reduced to much less and safer levels in the grains. So these can be popularized, and you can see that recently the genetically modified mustard plant has been developed by Professor Deepak Pental. So it has been now, it is now being grown. So you can see genetically modified plants are now being recommended by the environmental committees, the highest committee of the environment for the cultivation in the areas. So we are hopeful that these plants can be the plants can be also popularized. So I can summarize the topic, that the screening and selection and multi-location field trials with low grain arsenic cultivars resulting in development of the new rice variety known as Muktashri, which will be suitable for the growing in the arsenic contaminated regions. And you can see that we have made two amazing phytoclinic synthase gene and arsenite methyl transferase gene. And application of arsenic tolerant bacterial strains decrease arsenic uptake. Some of the bacteria which were having these arsenic methyl transferase gene, they were also they can use to be used in the rhizosphere. They can promote the PGPR activity, and they accumulated arsenic in the root zone, and very less arsenic. So through of some bacteria, or I can say the transgenic rice, where more and more arsenic can be accumulated in the roots, and less arsenic can be accumulated in the transgenic rice. So we showed that some of these transgenic, these phytoclinic synthase transgenic, and the arsenic methyl transferase transgenic from the fungus Westradicola is very useful. It can be used for making the transgenic phytoremediation, and you can see used for making the transgenic rice variety. We can decontaminate the arsenic loading in the plant, and as I told you that they can have safer level of arsenic suitable for consumption by the population. And this development of these, of course, it is a huge task, and it required a teamwork. So it required we had collaboration from different scientists. Dr. Devashish Chakravarti is a scientist here. Dr. Babut Kumar Trivedi is the director of the CMAP and the director of the NBRI also. Then Dr. Pankaj Kumar Srivastav, Pankaj Kumar Verma, and Shikha Verma, they made these these volatile transgenic rice plants using arsenic methyltransferase. Dr. Richa and Dr. Seema Mishra, they were in the phytoclatin synthase, and they make these transgenic plants, rice plants, transgenic of the phytoclatin synthase transgenics. And the Manjushri, Shikha Verma, Pankaj Verma, Richa Dave, she's very happy to see Richa, who helped me in the slide projections, and she did wonderful work. And she has done a very, very nice piece of work in her PhD program and identified the low arsenic contaminated rice genotypes, which have higher thiol levels and low arsenic levels. And in fact, this is the theory on which the phytoclatin synthase transgenics were made. Then the Garima Dixit, she is working in the Jaipur, and this PK Singh is working in the Medcare University. So my other student who worked on this rice, they are working in the Surbhi and Reshu Chauhan are working here. Dr. Poonam Tiwari has joined my group, and she is doing excellent work on how selenium can be used to prohibit the rice accumulation in the rice grain. And I think PK Singh and Dr. Poonam was getting some good data for publication and for having the rice variety with very low level of arsenic and high selenium. Selenium is very much required by the human beings. In fact, we require 59 microgram of selenium every day for the good growth of the hairs, nails, and many important, you have the selenoproteins. So we want to make using selenium, we want to make the low arsenic rice variety and high selenium rice cultivars. So with this, I come to end my presentation, and again, I am very much thankful to MIT University, Noida, Dr. Richa Dave, and other collaborators in MIT University, and IITR, who jointly gave me opportunity to do some of my work for the presentation here. So thank you very much. And well, I will be happy to answer if there are any queries or questions.
So it is actually great to revisit all the stalwart works that you have done, and our team is still pursuing on the lines. Is so heartening to see. So thank you so much, sir. And it is really a great learning experience, especially for me and all the participants.
Thank you very much.
Thank you so much, sir. Now, an announcement for the participants. All the participants are requested to fill the assessment form. The assessment link will be shared at the end of the session. It is compulsory for the generation of the certificates. Now, I would like to invite Dr. Richa Dave Nagar for the vote of thanks.
Thank you, Charu. We had a brilliant session with all the speakers who have come here and deliberated on different topics, and they have enlightened each and every of the participants with their vast knowledge today. In the 7th session, plenary session based on pollution prevention and sustainable solutions, Dr. Pratibha Kautam was the first speaker who spoke on pollution prevention and cleaner production, throwing light on the current condition of cleaner production and what are the specific requirements that are there and how the future holds for cleaner production in India. Then we had our next speaker, Dr. Manoj Chandra Garg, who had a hands-on training on virtual labs, and it was very useful for all our participants to gain from how they can use the virtual labs as practice or in their practicals. and the other online tools that are available. And even after the pandemic, the various online sources can be utilized to the fullest for more knowledge experiences. Thank you, sir. We have Dr. Vinay Kumar Khanna, Chief Scientist, CSIR IITR Lucknow, who represented the magic of quercetin as a neuroprotectant and the neurophysiological effects of cadmium. He spoke about the various aspects and covered different neurophysiological aspects and how quercetin can become a neuroprotectant. Thank you, sir, for this novel idea to be shared with all, and how we can have a brighter future with the innovations that you are bringing about. Thank you, sir. The last speaker of the day was Dr. R.D. Tripathi, sir, Nasi, Senior Scientist, CSIR, NBRI, Lucknow. We have been very blessed to have you with us, sir, and to be enlightened on the various aspects of arsenic toxicity and the mitigation strategies that can be used with rice, and for lessening the daily intake of arsenic through the subsistence diet of rice, the various biotechnological tools and the various new technologies and new breakthroughs that you have brought about bring a brighter future for all of us. Thank you so much to all the speakers. Thank you.
Thank you, Richa.
Thank you, ma'am.
So we can close the session now.
Okay, ma'am. Thank you.
Thank you. Thank you,
Tripathi,
Arvind
sir.
Thank you, thank you, Richa. How are you? I hope
I'm
everything
good,
is
sir.
fine.
Yes, sir.
Yeah.
So we'll be closing
It
the
was a-
session right now.
Yeah.
Yeah.
Generally, I don't say no to your invitations.
Thank you, sir. And it's always a blessing to have you.
Yeah.